എഴുതുകയാണോ? ഉണ്ടോ ഇനി എക്സ്ട്രാ? ഒരെണ്ണം ഇവിടെ വെക്ക്. ഇതല്ല, ഇത് ബാക്കിലുള്ളത് കറക്റ്റാണേ. ഇതിലുള്ള ആളെ കിട്ടാൻ വേണ്ടിയിട്ടാണേ. സെല്ലോ ടേപ്പ് ചെയ്യാം. സെല്ലോ ടേപ്പ് ഉണ്ടോ? അല്ല, ഈ സാധനം വെക്കാൻ രണ്ടാമത്തെ ആള് അതിൽ എഴുതണോ? അതിൽ എന്തിലും എഴുതണ്ട. അതിൽ എന്തെങ്കിലും അഡ്രസ് ചെയ്യുന്നുണ്ടോ?
അല്ല, അത് ആദ്യം പറയണം. വേണ്ട,
ഇത് വെക്കല്ലേ. അപ്പൊ സെറ്റ് ആയില്ലേ? സെല്ലോ ടേപ്പ് ഉണ്ടെങ്കിൽ ഒന്ന് ഒട്ടിക്കാം.
ഒരെണ്ണം ചെയർമാന്റെ കയ്യിലും ഒരെണ്ണം മറ്റേ ആളുടെ കയ്യിലും. ഇയാൾ ഇങ്ങോട്ട് നോക്കിയതാ.
അവർ അങ്ങോട്ടും ഇങ്ങോട്ടും എക്സ്ചേഞ്ച് ചെയ്യും. എക്സ്ചേഞ്ചിങ് ഓഫ് എംഒയു ആണ്. രണ്ടാൾക്ക് ഓരോ കോപ്പി വേണം.
ഒരാൾ കൊണ്ടുപോയ പോലെ രണ്ടുപേര് വേണം. അല്ല, ഒരാൾ കൊണ്ടുപോയി രണ്ടും കൊണ്ടുപോകണം. രണ്ടും കൊണ്ടുപോകണം. അത് ഫസ്റ്റ് എങ്ങനെ ഇരിക്കണം നോക്ക്. ഇതല്ലേ മറ്റേതിൽ ഇങ്ങനെ വെച്ചു. ഇത് കൊണ്ടാടണം.
This could be a good night for good times, and you look like a good time. We're dancing to dancing in the moonlight. A good night for good times, and you look like a good time. Dancing to dancing in the moonlight. Now I want you back. Can you make me feel better? Do you think you're better off alone?
guys ready to have a good day? This is my very first time in Biomarkers.
Smash the kick down, turn up the speakers. Turn up the speakers. Smash the kick down, turn up the speakers.
I thank each and everybody who have contributed to make this event a grand success.
Bloom 2025! Divine late Shri P.K. Das. Medicinal Garden is a 1-hectare land developed by utilizing the grant obtained from National Medicinal Plant Board. A grant amount of 5,40,000 rupees is obtained for the establishment and maintenance of garden for the time period of 5 years. Medicinal Garden is enriched with 250 plant species which are categorized under 16 subheading. The Herbal Museum of Pharmacy is a dedicated space that celebrates the richness of medicinal plants and their traditional uses. A key feature of the museum is the impressive collection of more than 50 herbarium frames, carefully preserved plant specimens mounted and labeled with botanical names, family, medicinal parts used, and their therapeutic applications. The lab provides a hands-on learning environment for students to identify, analyze, and understand the pharmacognostical and phytochemical properties of herbal materials. NSI 2025 was a dynamic interdepartmental event held on 16 January and 17, 2025 at Nehru College campus. This vibrant two-day celebration marked a unique collaboration among all the streams of the institution, including engineering, MBA, and pharmacy, bringing together knowledge, creativity, and innovation under one roof. The pharmacy exhibition, where students from different departments of pharmacy showcased their innovative projects, models, and research ideas. These exhibits reflected the scientific curiosity and practical insight of budding professionals. The Herbal Expo 2025 was a grand event celebrating the power and potential of medicinal plants, natural formulations, traditional remedies, and demonstration of different extraction methods.
Hello. A very warm and hearty good morning to one and all. We, the management, staff and students of Nehru College of Pharmacy, proudly welcome you all to the HERBOCARE 2025 one-day national seminar on revolutionizing the herbal healthcare biomarkers, the role of AI and ML in herbal drug discovery.
Once again, a pleasant good morning to a galaxy of intellectuals, teaching and non-teaching faculties, colleagues and students. It's our immense pleasure to welcome you all. Hearty welcome to the wonderful gathering from various pharmacy colleges.
No venture succeeds without God's blessings. Before we begin this program, in fond memory of our beloved legendary founder chairman, late Sri P.K. Das, and also former Chief Minister V.S. Achyutanandan, I humbly request all of you to please stand up to observe 1 minute of silence followed by prayer song.
ദൈവമേ ഞാൻ നിൻ സവിധേ കൂപ്പുകൈകളുമായെന്നും അണഞ്ഞിടുമ്പോൾ നീ നിൻ കൃപയെന്നിൽ തൂകണമേ. ദൈവമേ ഞാൻ നിൻ സവിധേ കൂപ്പുകൈകളുമായെന്നും അണഞ്ഞിടുമ്പോൾ നീ നിൻ കൃപയെന്നിൽ തൂകണമേ. സർവജ്ഞാനത്തിൻ ഉറവിടമാം ദേവാ നീ, നിന്റെ ജ്ഞാനത്താൽ എന്നെ നിറച്ചിടണമേ. ഞാൻ ചെയ്യും കർമ്മങ്ങൾ നിൻ കൃപയാലാകണമേ. എൻ്റെ വീഥികളിൽ നീ ദീപമായിടണേ. ദൈവമേ ഞാൻ നിൻ സവിധേ കൂപ്പുകൈകളുമായെന്നും അണഞ്ഞിടുമ്പോൾ നീ നിൻ കൃപയെന്നിൽ തൂകണമേ. കൃപയെന്നിൽ തൂകണമേ.
Before we go into the inauguration, let's go through our college. The Nehru Group of Institutions is a conglomeration of 20 plus institutions with 56 years of experience in the field of education. Starting from 1968, our founder chairman, late Sri P.K. Das, spent every ounce of his energy and sweat to establish various prestigious institutions in Tamil Nadu and Kerala. The brand NGI stands for Enriching Education Development with an emphasis on professional education such as pharmacy, nursing, medicine, engineering, etc. The vision and mission of the institution, building and matching the expectations of industry and society, are progressing apace, realizing the goals set.
The Nehru Group of Institutions among Nehru College of Pharmacy is one of the premier centers of excellence under NGI, involved in carving out disciplined, well-qualified and patient-oriented care of registered pharmacists willing to take up challenging tasks in industrial community and hospital pharmacy. The Nehru College of Pharmacy was established in the year 2003 by the founder chairman, late Sri P.K. Das, with the visionary zeal of serving the public of his home state, and has successfully completed 2 decades of service in pharmacy education. With constant effort of our management, NCP is recognized with NAC E-Grade Research Center with courses like M.Pharm in Pharmacognosy, Pharmaceutics, Pharmacy Practice and Pharmaceutical Regulatory Affairs, PharmD, PharmD PB, B.Pharm and D.Pharm. It's proud to say that now we are a UGC approved autonomous institution too.
Our mission is to emerge as a center of excellence that molds students into professionals and researchers who are socially committed with strong ethics and environmentally conscious. Once again, hearty welcome to all.
Professor Dr. K.G. Prashant, Principal in charge, Nehru College of Pharmacy, is a visionary who guides, motivates, and committed to providing a supportive and inclusive learning environment that fosters academic success and personal growth. I invite Prashant sir to deliver the welcome address. Welcome you, sir.
Good morning. Esteemed Chairman and Managing Trustee, Advocate Dr. P. Krishnadas sir, Honorable CEO and Secretary, Dr. P. Krishnakumar sir, respected Dr. H.N. Nagaraja, Executive Director, NGI, respected R. Gauri Madam, Director Research, Ranking and Accreditation, NGIK, beloved Principal, Dr. Vijay Kumar in his absentia, and distinguished guest, Nandan Kumar, and our speaker, Tirupati sir. My duty is to deliver the vote of thanks before going to the my duty. I want to brief about something about what is the motive behind this conference and why we organized such a conference. I thought that the history of humanity or human civilization and utilization of plants and natural products for both food and medicine to treat or prevent ailments has been, we don't know when it was started. And it is, what is mainly, when plants are, we have some literature that nearly 1,600 years ago, humans started cultivating plants and plants for food and medicinal uses. So, however, the significant concern regarding our alternative medicines or alternative therapy, even though we have the Asian books of Charaka was there, which explains about the uses of various medicinal plants and their properties and the uses of those medicinal plants for various ailments. But still we have some gaps in the treatment. When we found that there is the standardization of this alternative medicine or the therapy, whatever we are saying that the alternative medicine therapy, have a gap that what is the proof? That was asked by most of the English medicine people, as well as a lot of YouTubers are started asking, what is the role? What is the evidence? Where you are saying that this system of medicine is effective in treating so and so diseases. What is the proof? We are poor in documentation, and we are not at all documented properly, all those systems. So later, the alternative medicines are, once it became authenticated, or the therapeutic efficacy and the purity of the compounds involved can be identified or authenticated, then automatically, scientifically, if you are authenticated those things, this system should be accepted for the treatment. And mainly by identifying various biomarkers, like if suppose a Chavanaprasam or suppose any Ayurvedic preparation should contain a particular type of active ingredient at a particular level. That type of quantification we are able to do, then surely we are in the target. We are hitting the market. So still now, and we should be able to prove that by having such type of biomarkers or such type of active ingredient in such a preparations can decrease the level of sugar or cholesterol or enzymes, whatever it may be. That biomarker level we are able to measure and we standardize it, then our system of medicine will be accepted. And mostly if you can see that our the present government is working towards that, and especially Ministry of Ayush has established a GMP committee, and also we have an pharmacopoeia commission. We also have an Ayurvedic pharmacopoeia, which list out the active ingredient and the biomarker, what are the important ingredients present in each molecule, list out those traditional medicines. That gives us an additional value over the products. If you take about the, in our scenario also, if you see that Kerala scenario at this Karkataka Masam, we know that it is for Ayurvedic treatment. If you are able to standardize those things, and we say that after this treatment, these are the things which are the elements, these are the proving that these are the biochemical markers, or these are the biochemical things which will be changes happens in the human body, then our health tourism will be in a booming stage. It will, whatever we are having, a 50% hike will be there in our health treatment system, sector. So at present, we have 75,000 crores of export potential. Once, if we are able to standardize it, once we are able to prove that the system of medicine was effective, and these are the effect which can produce by our system of medicine, it may be go up to double. Nearly 15,000 crores will be the export potential of our system. That means a 20% growth, average growth per year. So, and therefore we are, that is why we are plan to conduct a conference, especially on the application of AI and ML, machine learning, to identify those markers, standardize the herbal products, and identify those markers and biological markers as well as therapeutic ingredients, so that we can standardize our products. So this will be an eye-opening for all the people who are, all the delegates who are sitting over here, and surely this conference will ignite a spark on the minds of the young delegates sitting over here, and you can also explore such areas in the near future, and you became an entrepreneur, a startup company. Anything can be, a seed can be started pouring here for such a big growth. So this conference may be an initiation for such type of, we can expect at least 1 or 2 entrepreneurs or 1 or 2 products has been developed after this, attending this conference, after something sparks on your mind, that we are successful in conducting this conference. So with this short introduction, I am coming to my duty. First of all and foremost, I would like to welcome our chief guest and resource person of today's function, Dr. Nandan Kumar D, founder, CEO, Innatura Scientific Private Limited. He's from Hyderabad. Welcome you, sir, for this wonderful conference. Thank you, sir. Next, it is my honor to welcome our esteemed Managing Trustee, Honorary Trade Commissioner to Mauritius, Advocate Dr. P. Krishnadas, sir, to this function. Welcome you, sir, for this function. I also extend the warm welcome to our respected CEO and Secretary, and P. Krishnamurthy, sir. Unfortunately, he is unable to attend the function due to some important meetings. However, he has conveyed his best wishes for the successful of this conference. Thank you, sir. I welcome Tirupati Boni, Research Officer, Department of Quality Control, Laboratory, National Ayurvedic Research Center, Panchakarma, for this conference. Welcome you, sir. Additionally, I welcome Dr. H.N. Nagaraja, Executive Director, NGI, to this inaugural function. Welcome you, sir. I would like to extend my warm welcome to Dr. R. Gauri, Director of Research Ranking Accreditation at NGIK, for this function. Welcome you, madam. I welcome all the delegates from various colleges throughout Kerala, Tamil Nadu, Andhra Pradesh, and Karnataka to this one-day conference. Thank you for all of you to come to visit to attend this conference, and hope that this will be a wonderful section for your wonderful section. I welcome all the invited guests of our sister concern, Dr. Marar, HOD, MCA, Computer Science Department, and our office staffs, Usha Madam, Benipul Sir, and also welcome our beloved Administrative Manager and Statutory Works, Mr. Srinivasan Sir, for this function. And I welcome all the HODs and my colleagues and my dear students for this function. And once again, I extend my warm welcome to all of them for this wonderful conference. Thank you.
Thank you, sir.
Light expels darkness. Defying gravity stands as a symbol of a quest for knowledge which enlightens our mind and soul. Across cultures and traditions, lighting a lamp holds a multitude of profound implications and resonance which symbolize the triumph of knowledge over ignorance, hope over despair, and light over darkness. Let's light the lamp of love and compassion. I invite all the dignitaries come forward to light the lamp. Welcome you all.
श्री भगवती समेत श्री भगवते नमः। ओम सच्चिदानंद परब्रह्म पुरुषोत्तम परमात्मा श्री भगवती समेत श्री भगवते नमः। ओम सच्चिदानंद परब्रह्म पुरुषोत्तम परमात्मा श्री भगवती समेत श्री भगवते नमः। सच्चिदानंद परब्रह्म पुरुषोत्तम परमात्मा श्री भगवती समेत श्री भगवते नमः। ओम सच्चिदानंद परब्रह्म पुरुषोत्तम परमात्मा श्री भगवती समेत श्री भगवते नमः। ओम सच्चिदानंद परब्रह्म पुरुषोत्तम परमात्मा श्री भगवती समेत।
Thank you all. Dr. P. Krishnadas, Honorary Trade Commissioner, Mauritius to India, Chairman and Managing Trustee, Nehru Group of Institution. A law graduate with Master's and Doctoral degree in Management, holds various official designation. He is a former Senate member of House and former President of Kerala Medical College Management Association. He is a recipient of various national and international awards like Multinational Business Excellence Award by Intimate Billionaires Club, Multi-Billionaires Award by Pegasus Global Private Limited, Man of Excellence Award, and more credits. Realizing the dreams of his father, he is a key motto to execute our very motto, molding true citizen. It's proud to see that he is the Chancellor of P.K. Das deemed to be University. With all due respect, I would like to welcome Krishnadas sir for the presidential address. Welcome you, sir.
Thank you. Good morning to one and all. Most respected Dr. H.N. Nagaraja, our Executive Director and the Vice Chancellor of P.K. Das deemed to University. Respected Dr. K.G. Prashant, who delivered the welcome address and vote of thanks to the welcome you. Respected our chief guest of the day, Dr. Nandan Kumar from Hyderabad, a scientist and a CEO and founder of a wonderful herbal company with a touch of modern medicines. Dr. Gauri Madam, Director of Academic and Administration Research of NGI. Mr. Srinivas, Dr. G.N. Pramodini Madam, Jasna, all other esteemed dignitaries on and off the dais, esteemed faculty members, other administrative staffs, and all other delegates or representatives from various other institutions and industries, teachers, once again, good morning to all. Today is a wonderful day to assemble this in hall for this HERBOCARE 2025, a one-day national seminar. It's going to be one of the best, challenging and interesting topic to all of us when it comes to the herbo and the medicine and other aspects. Already your beloved Vice Principal, Professor K.G. Prashant already explained what is going to be the point of discussions on today. In that itself, you now have the insight what's going to happen today. So according to the latest development in our country and across the globe, the importance of herbal medicines and the herbal ingredients are more, because already a lot of researchers already found out that it is having more value and more potential when it used in a proper way. And it's a modern era, it's a era of scientific era. So all research will be supported by the modern technology and intervention of the AI, machine learning, and even nano medicines and nano activities, and all other is giving importance to this kind of aspects. And in our country, when we talk about India, we have come out with a national education policy and also a new Ayush policy, and also the National Medical Commission also having aligned with the same thing. And all the Ayurvedic products and the pharmaceutical products, everything is now aligned, and it is almost in a singing stage or synchronizing stage. So, being the students or researchers of the pharmacy, especially this field, you have to understand what is going to be the tomorrow's issue. Because everything will be identified and invented, but later on, its after effects research will find out that something is missing and something is plus. So such kind of things to be debated. So for that, your vision and your thinking process should be in such a way, what all pros and cons will be happening when herbal involvement is seriously involved in the modern medicine. And when we talk about the Ayurveda, we know that it's a Karkataka Masam already mentioned, wherein a lot of Ayurvedic treatments are happening. Karkataka Kanji, that is a food, a staple, that is something different from other food, where it gives food, good nutrients and everything, it will be balancing our body, acidity, everything will be controlled. That means, that is the way how the ancient medicine has already evolved. And we know that we are the masters of that. Across the globe, even when we go to China, when we go to South Africa, or when we go to Korea, when we go to America, Europe, everywhere, wherever you can see, anything that is patent is only taken from India alone. Indian Vedic history, Indian science, for immemorial times and years and hundreds and thousands of years, they practiced this. All Buddha practitioners are already having this healing facility, and they are having the healing touch, everything based on this. So Bharat is a country where we are having rich tradition, culture and values, everything infused in our system itself. Unfortunately, we are not able to identify that so many years. Now it's identified and we are utilizing it. So it's a great, interesting topic. All the youngsters, those are sitting in the hall, don't think that we are completing the B.Pharm, M.Pharm program of PharmD or D.Pharm and we are going to dispense the medicine or we are going to be the industry. That is not the way. You have to be more and more energetic and thinking and how to use this kind of things in the modern medicine and how it will be curing the patients. And even in nearby, we know that it's surrounded by a lot of Ayurvedic treatment center. And even for oncology, especially cancerous cells are controlled with the latest Ayurvedic treatment. In some extent, even other alternative medicines are also used. So latest trend says, or the study says that everything should be used in a proper way with a proper research and proper formula that will be giving the correct output and the correct curing for any disease of humankind. Because we observe the nature, what's happening there. Everything is happening in the natural process. Nobody is taking care, but it happens in the natural way. So that is importance. Anyway, I am not taking much time because already it is too late to start the program itself. It's a one-day national seminar. Here everything will be discussed on all aspects, the chemical, the formulation, the marking even and many other things, the outcome, research, output, everything will be, the valuation, everything will be discussed. So I am not going to that. Moreover, I am not an authority on that. My knowledge and my involvement is only up to this because I am an administrator where I involved in medical, college, hospital and many other areas. Even I am into pharmacy. So I know something, that's all. I am not an authority like you. But one thing I know, what's happening in and around. I visited various countries, everywhere they demanded the requirement from that countries bring some Ayurvedic components over here which can be utilized and that can be served here. That is the saying of all countries. So that is important. So even in the medical profession, National Medical Commission has mandatorily infused an Ayurvedic program of 10 days for even for MBBS students. Even for Unani, even for homeo, even for other natural cure, everything is now combining together because the scripts and the manuscripts, everything says and tells like that. So anyway, I am not taking much time. My dear brother, it's a wonderful session. Definitely all the brothers and sisters involved and also participating in this program, especially the eminent personalities, those who are here from various colleges and organizations, you have to involve in detailed discussions. You have to debate with these people, the panel members, and find out, and all the aspects happening here should be answerable. Some solution should be there. Some real answer should be there. And the proceedings and the procedures of this one-day seminar should be answered to others. That should be a reference book for other colleges and students and researchers of our state and country. That should be the quality output to happen, that should happen from these proceedings. So my dear friends, I wish you a great day with this national one-day seminar, but involve thoroughly and ensure that you are also knowledgeable and wisdom person in this field. And people like here, especially Mr. Nandakumar and all, he's not a veteran, he's a eminent personality who studied abroad, who involved abroad and who worked in various organizations, now having his own company as a CEO and founder and doing wonderful things and a lot of R&D centers established in various institutions. And now he already morning he agreed that he is ready to have a collaboration with us and he will be giving an R&D output for us. So based on that we can work together. That's such a wonderful thing. So it will be benefiting you only. So take this opportunity as a golden opportunity. Ensure that whatever time spent in this hall should be fruitful and meticulously you follow the guidelines and protocols which will be helping you. With this note, I congratulate all the organizers behind this, especially I think Pramodini Madam and team and Prashant Sir and all other people, those who are involved in this. It's my great salute to them. Let this kind of program be answer to all other institutions. So let them also follow our steps and do wonderful things which will be helping the research and collaborations in our country. And we'll come out with more and more results and positive results in the country. Thank you. I wish everyone saying great day. Jai Hind, Jai Bharat.
Thank you, sir.
Dr. G.N. Pramodini, Professor and Head, Department of Pharmacognosy, having 18 years of experience in the field of pharmacy with nearly 35 paper publications, attended various national and international conferences, attained Best Paper and Oral Presentation Awards, and also formulated many herbal formulations. A person with dedication and unwavering commitment who always spent time, energy and passion to make this event a success. With immense pleasure, I would like to invite Dr. Pramodini Madam for Prelude to the conference. Welcome you, Madam.
Good morning, all. Myself, Dr. G.N. Pramodini, Professor and HOD, Department of Pharmacognosy, Nehru College of Pharmacy. This gathering I'm going to address with a small quote by Dr. APJ Abdul Kalam sir, like, life and time are the world's best teachers. Life teaches us to make good use of the time, and time teaches us the value of the life. So I welcome our Honorary Trade Commissioner, Mauritius, India, and Chancellor P.K. Das, deemed to be University Chairman and Managing Trustee, NGI, Dr. P. Krishnadas sir. I welcome our respected P. Krishna Kumar sir, Pro-Chancellor P.K. Das, deemed to be University, CEO and Secretary, NGI, in his absentia. I welcome all dignitaries, Dr. E.H. Nagaraja sir, Gauri Madam, and Dr. Vijay Kumar sir in his absentia, Principal, and I welcome our Vice Principal, Dr. K.G. Prashant sir, and I welcome Mr. M. Srinivas sir, who is the Administrative Manager. So I welcome our Chief Guest, Dr. Nandakumar Dudukuri, Founder or CEO in Natura Private Limited Company. Sir has come all over from Hyderabad to address the gathering, and he is the Chief Guest. I welcome our eminent speaker, Mr. Tirupataya Boini, who is the Research Officer, Department of Quality Control Laboratory, Ayurveda Research Center, Panchakarma. And I welcome all the eminent scientists, respected faculties from various colleges, industry professionals, dear participants, and my dear students of Nehru. We extend, on behalf of staff of NCP, a gratitude to the management for their unwavering support in organizing this seminar, HERBOCARE 2025. This event would not have been possible without the constant encouragement and facilitation from our respected management. We are deeply thankful to the management for providing the necessary resources and infrastructure for this seminar. The seminar mainly reflects the vision, commitment of our management towards academic excellence and knowledge sharing. We are mainly inspired by the leadership of our management, which constantly motivated us staff for innovations and growth in research. It is with our great enthusiasm that we, the Department of Pharmacognosy, Nehru College of Pharmacy, welcome you to the Pioneer Conference, HERBOCARE 2025. That is, Revolutionizing the Herbal Healthcare Biomarkers, the role of artificial intelligence and machine learning in herbal drug industries. This conference brings together pharmacy faculties, HODs of various disciplines, students, researchers, technologists, and traditional medical experts on a single platform for exploring their dynamic intersection and ancient traditional wisdom and modern innovations. As well as, we all know from centuries that traditional herbal medicine has played a vital role in healthcare system across the world population. Even WHO has recognized these herbal medicines that can be given as practitioners. And today, with the rise of the AI and ML, we are witnessing the massive transformation shift in how we can explore, validate, and develop the herbal remedies into herbal drug formulations. These sophisticated tools, that is AI and ML, hold immense potential to accelerate the phytochemical analysis, identify novel bioactive compounds, optimize the formulations, and predict therapeutic outcomes, all with an unprecedented speed and accuracy. This conference's main aim is to serve as a catalyst for interdisciplinary collaboration, sparking discussions and discoveries that will drive the next generation of herbal drug development. Through expert talks, technical sessions, and case studies, we will dwell into how data-driven approaches can revolutionize the traditional knowledge and integrate into evidence-based herbal medicine. The conference will be benefiting all the participants with insight knowledge about the herbal drug formulations with our best practices. We are also signing an MOU with Inauguration Scientific Private Limited, which will help to bridge the gap between the academia and herbal drug industry by providing training on sophisticated equipments and giving placements and guidance to our students, as Inauguration Scientific Committee have placed many of their trained students in various reputed pharmaceutical companies across India and abroad. We, the Department of Pharmacognosy, extend our sincere thanks to all the eminent speakers, contributors, co-sponsors, and attendees who have supported and participated. Participation is made this event possible. Your presence has made a shared commitment to innovations and herbal drug formulation, sustainability, and global health. So let us embark on this exciting journey together, where traditional knowledge blends with the technology and creates the limitless possibility in herbal drug development process. Thank you one and all, and let the conference begins. Thank you.
Thank you, ma'am.
Dr. Nandan Kumar Dudukuri is a distinguished scientist, entrepreneur, and academician with a remarkable career spanning drug discovery, pharmaceutical research, and global business development. Currently serving as a CEO and Chief Scientific Officer of Inauguration Scientific Private Limited. He is at the forefront of building innovative B2B networks across Europe and North America. His company specializes in providing comprehensive drug discovery services. He is a young scientist and principal investigator under the prestigious SERB Startup Research Grant, and many more in his credits. It's our pleasure to get such an eminent personality today. I invite Dr. Nandan Kumar sir for the inaugural address. Welcome you, sir.
Good morning, one and all. And first of all, greetings from Inauguration Scientific. I'm very happy to be part of HERBOCARE 2025, along with the dignitaries, Dr. P. Krishna Kumar Krishnadas sir, and Dr. P. Krishna Kumar sir, who is continuing the legacy of Dr. P. K. Das, and Administrative Director Nagaraj, and the Scientific Officer Tirupataya Garu, and scientific community, and my dear students. Here we are celebrating herbal medicine and ancient Ayurvedic and Siddha innovations and inventions of Indian community. And in India, there is so many of natural products available. Abundant herbal resources are available, like not only in India, China, and Thailand, but we are utilized properly, been from decades, and supported, and simply we can say it's a practice. Ayurvedic is not only medicine, it's a practice. It's not a new term for us. We are already been practicing. We are, while eating food, we involve with the almost medicinal plants. But in abroad, when I visited South Korea and China, they don't have that kind of herbal resources. Of course, China has, but in English country, in Western countries, they used to go with some whatever the processed food, ultra processed food. But why they use means like they don't have the knowledge, awareness regarding herbal importance and herbal medicine and natural resources they don't have. So we are very lucky, but we are not able to make the branding or maybe give the awareness and making the products to support our health and for healthy lifespan. Because we have to observe not only herbal medicine or maybe herbal industry or maybe natural product nutraceutical industry. We have to observe both the pharmaceutical and herbal industry, how these two areas are emerging, how they are branding, mostly pharmaceutical, Western medicine, English medicine, how they are branding, how they are marketing, how they created the culture. But in natural products, while dealing with technology or maybe producing any product or dissemination of the information and going with the market, we are missing many things. First, we have to create the culture. The students are more suitable to disseminate the information regarding this, whatever the HERBOCARE we are trying to explore and trying to give the knowledge and support the herbal medicine. And another thing is, I would like to suggest students you have to improve the curiosity, critical thinking, and creativity while going through your studies. Here in industry or maybe any research center, in any abroad university, they will see your skills. They want outputs. They have the deadlines. But we have to develop the skills and we have to develop the curiosity. We have to position well. Positioning is very important. Before that, we have to identify which area is very suitable. Try to interact with the industry people. Try to interact with the research centers. Meanwhile, while completing your curriculum, try to get some mentor first. Means like where you are trying to, example, cognosy. So in cognosy, if you would like to be one of the herbal medicine producer, maybe you want to be an entrepreneur, try to get the mentor first and try to get the knowledge. We also got mentors. We have the mentor from, he supports senior management team of Hetero Drugs. He trains and motivates the senior management. He is mentor for me. That's where I always go with him. If I got any critical problem or maybe critical discussion with the client or maybe with the partnership, simply I'll ask him. He will give only one sentence or one word answer. So that's where the mentors are very important while leading your career. One of the example I can say, in our company, we recruited one fellow from village background. He don't have the, proper, we got a qualification in MSc Organic Chemistry, but he got the backlogs 3-4 years back. So he was not willing to join the company, but his father is willing to push him to go with the industry. I said one word to his name is Raghu, Mr. Raghu. So I said one word, Raghu, if you are staying in village, going with agriculture, I don't think so. That is one of the block to limited your lifestyle. If you come to Hyderabad, if you stay, whatever we are giving the training for 1 year, 2 years, then it would be very, then you establish in pharma industry, you can establish your family in Hyderabad. Maybe you may lead towards career towards abroad, I said. So initially he was not willing to join. But because I heard the sentiment that he is from my place only and my BSc college. Then after 1 week he called and his father also pushed him and he joined. After 18 months, after almost around 2 years he worked with us. Later he joined in some clear scent company. It's very good company making impurities. Within 4 months he got best employee award. You don't have soft skills, you don't have any theoretical knowledge regarding the work, but he is the focus. When he give the work, he is able to work and he able to give that proper time, and he got professional ethics. So, and now he is leading team. Within 4 months he support for 16 towards market product. His team leader got 12 lakhs car as a bonus. He got 10,000 rupees of incentive. You see, like he is going to be a failure. He don't have the during Corona time we recruited him actually. Like that, anybody can be lead the career, but positioning is important. If you are want to be a student lifetime, just go with the curriculum. But if you want to lead the field or maybe lead as an entrepreneur, you want to go through the big companies, MNCs, you have to link many things. Networking is very important. Communication skills are very important. Maybe you don't have the soft skills. You don't have the maybe skill set regarding what you are going through. And if you go with the multitasking and trying to explore the areas and try to connect with the many people and don't isolate. I'm suggesting go with some friends and try to share your ideas, and who are good at giving the good suggestions. Try to share your skills. They will tell you, this skill is good. You are good at whatever you are doing this thing. Then you can excel. Even my friends many times encourage me during PhD. And then I got this, opportunity to be, and I got an idea to be an entrepreneur. Now I am here. And many things we have to discuss technically while giving the presentation. And thank you very much for the opportunity. Yeah.
Thank you, sir. Dr. P. Krishnakumar, Pro-Chancellor, P.K. Das Deemed to University, CEO and Secretary, Nehru Group of Institution, a man with leadership who demonstrates strong qualities that enable him to guide and inspire us. With immense pleasure, I would like to welcome Dr. P. Krishnakumar, sir, in his absentia. He already conveyed his regard for the success of this event. Thank you, sir. Dr. R.C. Krishnakumar is the Director Operations at P.K. Das Institute of Medical Sciences. Due to his busy schedule, sir is also conveyed his regards for the success of the event in his absentia. Thank you, sir. Professor Dr. H.L. Nagaraj, Executive Director, Academics and Administration, Nehru Group of Institutions, distinguished academic and administrator with extensive experience in education and research, and we can proudly say that he is the Honorable Vice Chancellor of P.K. Das Deemed to be University. I invite Dr. H.L. Nagaraj, sir, for the keynote address. Welcome you, sir.
Very good morning to all of you. Respected President of today's function, Honorable Chairman and Managing Trustee of Nehru Group of Institutions, Honorary Trade Commissioner of Mauritius and India, Chancellor of forthcoming P.K. Das Deemed to be University, Coimbatore, our beloved Dr. Krishnadas ji, Chief Guest of today's function, Dr. Nandan Kumar Duddukuri, Founder and CEO, Intura Scientific Private Limited, Hyderabad. Guest of honor and the resource person, Mr. Tirupataya Boini, Research Officer, Department of Quality Control, National Ayurveda Research Institute for Panchatantra. Respected Director of Accreditation and Ranking, Dr. Gauri Madam. Respected Vice Principal of Nehru College of Pharmacy, Dr. Prashant, Dr. Pramodini Madam, Mr. Srinivas, the Administrative Officer of NJ Kerala, Professors, Directors, Deans of Sister Concern Institutes and Nehru College of Pharmacy, delegates who have come all the way from various institutions of different states, and my dear own student friends. At the very outset, I first of all congratulate the College of Pharmacy for having hosted this particular national seminar on a right topic. Friends, why I'm telling right topic is, within your 4 years study, what you are going to study is already the bookish printed information which is there available in the book. As far as artificial intelligence and machine learning all are concerned, this is the technological development which is happening day to day. We cannot incorporate everything day-to-day changes in the books because we are affiliating to some universities, and though we are having autonomous status, so we cannot immediately implement. And we cannot make you to teach or make you to learn everything so within the stipulated time. That's why quite often these type of activities will definitely throw limelight, particularly in the advanced technology, what's happening outside. Friends, as far as technology is concerned, it is developing very fast, particularly in a country where we are fortunately born, that is India. It is growing so fast. And in economic sector also it is moving fast. In every sector, whether it is health sector, space sector, agriculture sector, so and other industrial development as such, in every sector India is moving very fast. And the contribution is by your own brothers, sisters, parents who are working round the clock throughout the globe. And as far as coming to the technology and its impact in the health sector is concerned, say generative artificial intelligence, it's playing a prominent role wherein all the information, the data of a patient is being collected, analyzed, and what actions is to be taken is being carried out. Earlier, it was little difficult for the doctors and the people who are looking after. But all the information through artificial intelligence, it is possible nowadays. Similarly, so telehealth. So telehealth is nothing but teleconference in the form of audio conference, video conference. A doctor sitting at California can immediately not only guide the doctor, so present here, he can even so get the things done so through video conference. Nowadays, one step ahead, he need not require a man, so he requires even a robot. A doctor sitting at California can get the patient operated, so surgical activities through a robot which is present here. So that much technology is going on, particularly in telehealth as such. Similarly, nanomedicine, as our Honorable Chairman has told, so nanomedicine is also invading very fast. So nanoparticles, that is, so obstacles are the materials. Say for example, nanorobot. Nanorobot is very, very small, made to flow or made to enter, pass through the blood vessels. Blood vessel itself is a very small. So through blood vessels, it is made to pass through, and it can easily identify the cancer cells. It can easily identify the virus present in the blood cells and all. So like that, nanorobot is also there. Similarly, so robots are also being used for chat robots and virtual assistants. Doctors most of the time requires assistants, particularly say for example, I am coming from Karnataka, I cannot speak Malayali. If I have to consult with doctor, if I do not know even English and all, I have to speak in Kannada. Then doctor is unable to understand. Under such circumstances, this chatbot will play a prominent role. This chatbot will convert the language, so corresponding to the patient's language, what he is speaking, it will convert and it will deliver to the doctor. Even it prepares the notes. Even it prepares the notes and gives it to the doctor, so it will work like literally an assistant like. So like this, the technology is developing and helping particularly the health sector people, so in a very wide manner. Coming to your own pharmacy, say for example, I go to a shop and I ask for a medicine, which medicine, where it is kept and all, it is not known to the person, so he will start searching. So artificial intelligence plays a prominent role in identifying, so because it knows where it is, and so when it is kept, even the stock, how much stock, he ultimately after searching for 10 minutes, he says it is exhausted, it is not available. If AI is utilized, it can immediately tell that whether it is available or not. Like that, artificial intelligence, not only for fabrication of the medicines, even what are the components to be incorporated, how much, whether it is expiry date is there or not, everything artificial intelligence will be helping for the doctors in this. And similarly for the robots can also be utilized for delivering of the medicines. Delivering of the medicines, chatting with the patient, everything is possible. So artificial intelligence is playing a prominent role. So these things, now stalwarts have come all the way from different places. So if you listen literally from them, it will throw a limelight and it will give you more exposure particularly. So with this, without taking much time, I only wish the entire one-day seminar will go a long way, and all of you who have come particularly from outside and all, you will take the best out of this seminar, and afterwards also you will be in touch with the resource people so as for your better benefits, and I wish the seminar goes well. All the best, and wish you all the best. Thank you, Ananda.
Thank you, sir. Dr. R. Gauri, Director, Research Accreditation and Rankings, a prominent educational conglomerate who oversees the institution's research initiative, accreditation process and ranking strategies, contributing to NGI's commitment to academic excellence and innovation. I invite Gauri ma'am for keynote address. Welcome you, ma'am.
Very good morning to one and all. Respected Chief Guest of today's function, Dr. Nandan Kumar, Guest of Honor and invited talk, Research Scientist, Research Officer, Mr. Tirupataya Boni. Hope I spelled both of your names correctly. If not, Sorry for it. Honorary Trade Commission of Mauritius, India. Respected Chancellor P.K. Das, deemed to be University, Tamil Nadu. Chairman and Managing Trustee of NGI. Dr. P. Krishnakumar, CEO and Secretary. Pro-Chancellor of P.K. Das, deemed to be University, in his absence here. Respected H.N. Nagaraja, Sir, Executive Director of Academics as well as Administration of NGI. Vice Principal Dr. K.G. Prashant, Sir. Convener of this national seminar, Pramodini Madam. Faculty and staff members of Nehru College of Pharmacy. The person behind the success of and promoting the activities of Pharmacy College to the greater extent, Srinivasan Sir. Participants, delegates from various states of country, and my dear students. What as a convener Dr. Pramodini Madam has told to me, conveyed to me, let me tell very clearly that 17 to 18 colleges have given their contribution and around 300 participants have done the registration for this conference. Let me take the privilege as a director of research. In a span of 2 months, we are able to attract 300 participants and more than 100 presenters will be there. That is, their abstracts will be getting into the seminar and it will be given. So, we can imagine the demand of this topic across the nation and across the globe. Because the role of AI and ML has produced the complete evolutionary aspects in all fields. I'm not the person to talk more about pharmacy terms and all, but I can say that use of machine learning and AI make use of the large samples of data which we as a human being cannot do it. Because if we go for collecting the samples, maybe we may get hundreds or maximum. Some hospitals in and around will take and collect the data and everything. But AML gives the predictive analysis by making use of huge data being stored in the cloud. So that process, and it saves the time and gives the more accuracy than that of the olden days. So this conference is a multidisciplinary one. It is not interdisciplinary, it is multidisciplinary, which our nation is giving a great focus onto it, interdisciplinary and multidisciplinary. Because AML, the people think that it is computer science or computer engineering related, and of course, HERBOCARE from the herbal products, medicines, so drug discovery, because some people will have the fear that, okay, allopathic medicine, every time it will keep on increasing the dosage and our immunity of the human body system will keep on reducing. So the people, sometimes we go with, okay, let me try with homeopathy, let me try with Ayurvedic and various medicines, right? So the accuracy and the satisfaction can come only with the data analysis proving. Because today's era is not like a blindly accepting anything. So this conference will give you, this conclave will give you the platform to share your thoughts. Please, in one day, all the people cannot communicate, but through the convener might be having the records of contact details of each and every participant. So make use of it and let us explore the system as a continuing platform for the collaborative research. With these words, I wish you all the best and grand success to this event. Thank you all.
Thank you, ma'am. I invite Mr. M. Srinivas Nair, Administrative Manager, Nehru College of Pharmacy, a person with infectious energy and lively spirit who always motivates and inspires us for the felicitation. Welcome you, sir.
Thank you. Most revered and respected Chairman and Managing Trustee, Advocate Dr. P. Krishnadas, Sir, Honorary Trade Commissioner of Mauritius, India, Chancellor P.K. Das, deemed to be University, the leader of Nehru Group of Institutions who leads us from the front, the Honorable guest, Chief Guest, Dr. Nandan Kumar Dudukuri, Founder and CEO, Innatura Scientific Private Limited, Hyderabad, the Guest of Honor, Mr. Tirupataya Boini, the Research Officer at Department of Quality Control Laboratories, National Ayurvedic Research Institute for Panchakarma, Cherudurti, the Honorable Executive Director, Academic and Administration, Professor Dr. H.N. Nagaraja, Sir, Dr. R. Gauri, the Research Director, Accreditation and Ranking, NGI, the Honorable Vice Principal of Nehru College of Pharmacy, Professor K.G. Prashant, Dr. K.G. Prashant, the dignitaries, the convener of this wonderful HERBOCARE one-day national seminar, and powerful delegates which have come from almost 15 colleges across Kerala. A warm good morning. It's already late. We are running short of time. I see it in a different perspective. Nehru College of Pharmacy, is having its own herbal garden and herbal extracts and herbal products. This is one of the best practices we have been following over the years with the support of our herbal garden, and which is supported by the National Ayush, Department of Ayush. We are making inroads into the products and its development. I am extremely sure that this HERBOCARE will stand with the time and be a grand success and discuss various modalities and various research opportunities in this field. I wish it thunderous success. Thank you.
Thank you, sir. In this moment, it's great to say that Nehru College of Pharmacy and Innatura Private Limited has signed a MOU. We are truly honored to have you all join us as we embark on the new chapter of collaboration. I invite Advocate Dr. P. Krishnadas, sir, Chairman and Managing Trustee, along with Dr. Nandan Kumar, CEO and Chief Scientific Officer, Innatura Private Limited, to exchange the MOU. Welcome you. All the dignitaries, please join. On behalf of Nehru College of Pharmacy, I invite our chairman, sir, to hand over a memento and certificate to the resource person, Dr. Nandan Kumar, as a token of our love and gratitude. I invite Advocate Dr. P. Krishnadas, sir, to hand over the memento to the resource person, Dr. Nandan Kumar, as a token of our love and gratitude. Also, we are adorning our chief guest with Pondada. We have one more resource person, Mr. Tirupataya Boini, Research Officer, Department of Quality Control Laboratory, National Ayurveda Research Institute. On the behalf of Nehru College of Pharmacy, we are providing a memento and Pondada as a token of our love and gratitude. are the first college conducted faculty development program under KUHAS, and we got an appreciation certificate from Kerala University of Health Sciences. Already our chairman sir received it from the university. Now our chairman sir is handovering the appreciation certificate to Dr. K.G. Prasad, Vice Principal, Nehru College of Pharmacy. It's great to say that we are the one and only college who have conducted FTP under KUHAS. Thank you all.
I would like to invite Mrs. Jisna TG, Associate Professor, Department of Pharmacognosy, to deliver the vote of thanks. Welcome you, ma'am.
Good morning, everyone. Myself, Jisna TJ, Associate Professor from Department of Pharmacognosy. It's my great privilege and honor to propose the vote of thanks on the special day of one-day national seminar, HERBOCARE 2025. On behalf of the Department of Pharmacognosy Organizing Committee, I express my gratitude and thankfulness to Honorary Trade Commissioner of Mauritius, India, Chancellor P.K. Das, Dean to be University, our Chairman and Managing Trustee, NGI, Dr. P. Krishnadas Sir, for providing all support and resource for initiating the National Conference HERBOCARE 2025. Thank you, sir. I would like to thank Dr. P. Krishna Kumar, Pro Vice Chancellor, P.K. Das, Dean to be University, CEO and Secretary, NGI, in his absentia. I extend our heartfelt gratitude to our Chief Guest, Dr. Nandan Kumar, Founder and CEO in Natura Scientific Private Limited, for taking time out of your busy schedule to be with us today. Your presence and your thought-provoking address have set a brilliant tone for the entire event. Thank you, sir. A special thanks to our eminent speaker, Dr. Tirupadaya Boini, Research Officer, Department of Quality Control, National Ayurvedic Research Institute for Panchakarma, for coming here to share his knowledge, experience and insights. Thank you, sir. I express my thankfulness to Dr. R.C. Krishnakumar, Director Operations, PKT IMS, in his absentia. I would like to extend my heartfelt gratitude to Dr. H.N. Nagaraja, Vice Chancellor, Executive Director, Academic and Administration, NGI, for delivering the enlightening keynote address that set the tone for our event. Thank you, sir. Our sincere appreciation also goes to Dr. R. Gauri, Director, Research Ranking and Accreditation, NGI, for sharing her valuable insight through the keynote address. Thank you, madam. We would also like to extend our gratitude to our esteemed principal, Dr. D. Vijay Kumar, whose vision and leadership guided us even in his absence today. Our heartfelt thanks to Dr. K.G. Prashant, Vice Principal, for his unwavering support and guidance throughout the journey of HERBOCARE 2025, making this national seminar a resounding success. Thank you, sir. I express special thanks to our Administrative Manager, Mr. M. Srinivas Sir, for providing endless efforts and guiding us in this national seminar process. Thank you, sir. I sincerely thank our IQSC Director, all the heads of department and professors, as well as the teaching and non-teaching staffs, our students, NSS volunteers for their invaluable support that made this event a grand success. Thank you all. We extend our heartfelt gratitude to Mr. Benny Paul, Administrative Officer, and our beloved Mrs. Usha Madam, Office Superintendent, for their invaluable support and assistance. Thank you. Special thanks to Mr. Govindaraj, Assistant Superintendent, for his tireless support and diligent follow-up, ensuring the smooth execution of this event. Thank you, Govindata. I would also like to thank our respected delegates, researchers and participants who have joined us from various institutions and organizations. We extend our sincere gratitude to the principals of Ahalya College of Pharmacy, Karuna College of Pharmacy, KTN College of Pharmacy, Maulana College of Pharmacy, Al-Shifa College of Pharmacy, Sri Ramakrishna Institute of Paramedical Sciences, Sanjo College of Pharmacy, Daleview College of Pharmacy, CPS Government Medical College Kannur, Corps Government Medical College, Elims College of Pharmacy, and all the other participating colleges from South and North India who encourage their faculty and students to be a part of this national seminar, enriching it with their presence and participation both online and offline. Thank you all. Our sincere appreciation goes to our co-sponsors, CO Pharmaceutical Private Limited, to support this event. Behind every successful event is a team of hard-working individuals. A big thanks to all my department organizing committee, our HOD and Professor Dr. G.N. Pramotini Madam for her continuous support, and my fellow staff members, Mrs. Drushya, Assistant Professor, Mrs. Sri Lakshmi, Assistant Professor, our second and fourth SEM M.Pharm Pharmacognosy students. Thank you all. We are thankful to all technical staff and logistic team for their dedication, planning, and seamless coordination for the event. Finally, I thank each and every one of you for being a part of this conference and for contributing to its success. We hope the session will be informative and inspiring, and we look forward to continued collaboration and knowledge sharing. Thank you all once again. Have a great day.
Thank you, ma'am. Now the official inauguration of the event is over. I request the presence of all dignitaries of the dais as we proceed by the session of resource person. Before that, just all of you please raise up for taking one photograph. I request the presence a testament to his unwavering commitment to advancing pharmaceutical sciences. He began his career as a young scientist at the Department of Pharmaceutical Technology, and he has also visited the Michael Bishop Institute of Cancer Research, China, and contributed to cutting-edge cancer research. In addition to this, he holds a scientist MBA from the USA and a postgraduate diploma in intellectual property rights from IGNOU. He served as an assistant professor at the College of Pharmacy, Gakon University, and the Lee Hilgya Cancer and Diabetes Institute, South Korea. Also completed a prestigious postdoctoral fellowship at Department of Nuclear Medicine, Samsung Medical Center, South Korea, and earned his PhD in synthetic organic chemistry from CSIR, Indian Institute of Chemical Technology, Hyderabad. Throughout his career, he has been recognized for his outstanding contributions to science and research. He is a young scientist and principal investigator under the prestigious SCRB Startup Research Grant and many more in his credits. With immense pleasure, I invite Dr. Nandan Kumar sir for the scientific session. Over to you, sir.
Hello, how are you all and good morning. All the way from Hyderabad to Kerala. This is my first visit. I really enjoyed the journey. Till the session starts, I felt it's a tour, not a scientific session. I really enjoyed the journey. The nature is very good. I think you know the Hyderabad biryani very well, right? Everybody enjoys in India and worldwide. And also Hyderabad is famous for, Hyderabad is a pharma hub and now it's life science hub. That's where we have the abundant talent pool and knowledge related to pharma and life sciences and also related to herbal medicine. As we are celebrating the Herbal Care 2025, here we are, it's like when I got call from the faculty regarding the lecture. So it's related to natural products. You know, mostly what are the people invited me 50% from the natural product laboratories and herbal medicine. I invited by China, Chengdu, the bio hub during before Corona. So that is also 2,000 acres botanical garden. To start that, I was one of the invitee. So here, the title is, like that time I decided, giving about the natural products, the knowledge already we have, the exposure we have already. So what could be the title and the topic to discuss and explore and get benefited by the audience and the scientific community and students. I thought, drug discovery, that the term is related to almost with the pharma industry's English medicine. But it's specifically, it's happening, it has so much of market, and you know very well, AI tools. I think everybody knows AI tools, yes? So we can say more than AI tools, these are AI agents. Now we are dealing with the agents, not only tools, right? So the agents do multiple things. The tools we can specifically use for certain part of the work. So I thought this could be the suitable title to go with the seminar. So natural drug discovery and smart molecules and smart methods. So I felt that the molecules are everywhere. Molecules are in natural products, molecules are in pharma, in tablet, or maybe in maybe any material, simple material that is in a plastic chair. It's a molecule made by molecule. Whatever the stage, everything is with having some molecule. So our in natural scientific is basically we do service wherever the chemical technology involved, we are happy to support. So we do specialty chemicals, fine chemicals and natural products. So That's where smart molecules came. Why I'm telling you smart means these molecules are mostly druggable and having applications. As per my affiliations and my knowledge related to this topic, here I would like to start. So here is the contents, introduction of the topic on source of natural drugs, strategies used for the drug development from natural products, AML in natural product drug discovery, how it can apply for natural product drug discovery, smart molecules and timelines, smart molecules, various smart molecules we'll discuss, and biomarkers, herbal healthcare. You know, herbal healthcare, like, the biomarkers. You might have the visited hospitals sometimes for MRI scan, CT scan. Those people, mostly that's highly applicable. That is biomarkers only. You will take some, they will give some drug actually for while going before the CT scan, right? So after taking that, drug, they will give, I think 2.5 liters, 2 bottles of water or maybe juice to excrete immediately. Otherwise you will get burning sensation. There is a drug actually with that is with the biomarker. So we'll discuss things. And applications of AML in herbal drug discovery, A and drug discovery, real world success stories, regulatory affairs related to how to excel in herbal medicine, challenges, strategic solutions, future perspective. So here, what exactly basics of the natural products, how we are doing classical approach, how you collecting the natural products or maybe herbal materials. So source is, first is plants, animals also, like even snakes also, snake venom, and microbial resources. Most of the microbials having that natural resources which are having cytotoxicity towards most of the cancers. And mineral sources, animal sources, we can say one of the things is like that is marine biotechnology, marine natural products. So from the sources from sea or maybe in the water, like what we can say, there is a plants also and animals we can extract. Yeah, here is we collect the plant from the we'll try to collect the plant whatever we decided, certain plants like Punarniva, that's name is, scientific name is Trianthema decandra. We isolated and we extracted and we supported for some US team, research team. We collect the plant and we'll try to identify exact name of the plant, and we have to confirm this plant name by any botany scientist or botany professor. We used to confirm that name because simply if you can find in Punarniva, Trianthema anywhere in like in the park or maybe in some places, it's kind of weird. So but we don't know, it's having 2-3 types of Punarniva, like white flowers, it would be like red flowers, but we don't know exact name. So we have to certify that with the botanical scientist or maybe botanical professor. And when we are working with the NPL division in IACT, CSR IACT, we used to visit with the plant before, after collection of the plant, some sample will take to the botanist professor, and they will identify the plant and they will give the exact name, whatever we are thinking is the same or not. Then then they will store in herbarium. It's a big herbarium is available in Osman University. So then we'll get the herbarium certificated, that is authenticated, that is the name of the Trianthema decandra, whatever you expected. Then we'll go for the extraction of that plant. Before the extraction, we try to make the powder, sometimes share dry, and sometimes we'll make some solution kind of to extract, and we'll go for the isolation. That extraction we do by using some organic solvents, sometimes water. That depends on what we are expecting. Sometimes we expect alkaloids, some terpenoids, some exact molecule which is already isolated or which is available and which is applicable for certain disease. Otherwise we have to identify some, we have to do some deep research, higher R&D, that is unknown molecule we would like to extract or isolate. So that by that we decide the solvent system, high polar solvents and low polar. when I was doing PhD, I did column chromatography of the natural product. It's a big column would be there, and I worked for 1 year on that same only one column. That is plant is highly poisonous plant is further history for us. That time we started with the pet ether, petroleum ether is hexanes, is highly low polar, not this low polar completely. From low polar to ethyl acetate, DCM, methanol, water. It took almost 9 months to 1 year. So that is extraction we do, and fractions we'll make, and we'll again do get the fractions, and then we'll go for the isolation. And after isolation of certain molecule, we do characterization also, structural isolation. If it is available, the molecule is a known molecule, we'll compare with the standard, and we'll get the NMR data and HPLC data, purity. We'll check mass and everything, 2D NMR if you need it, we take. And check the literature regarding the molecule background, and we'll go for the decide the bioassay, which bioassay would like to do, antimicrobial or maybe anti-cancer, anti-diabetic. And then we'll, after getting preliminary data, we'll go for the lead optimization. This is regular natural product drug discovery, what we do. Here is you can see the pseudo-natural products. Most of the natural drugs are, almost like 40% of drugs are from natural resources only. More than 60%, sometimes natural and analogs of the natural products are maybe pseudo-natural products. Pseudo-natural products, you can say, that is you can see eulin and morphine. This is they identified some structure which is indole structure and his morphine structure. They combined and they designed new endomorphine. Like that, molecules many are utilized in the pharma, English medicine. Most of them, like 50% above. So, but we'll see that is English medicine, pharmaceutical research and pharmaceutical products. But 50% are from natural resources, guided by and inspired, motivated from the natural products. You can see cyanic acid, ventamycin, that is a pyrrole pyridine ring, and they combined, they made pyrrole quinoline. So like that, many molecules are available, that is piperidinothiazole, pyrrolidone, methanol. Yes. So here is our work when I was doing PhD. He is my PhD guide, Dr. Biswanath Das, FRSC. He was a pharmacy chief scientist in laboratory. He is around 400 plus publications he got in natural product isolations. He is a very famous scientist in India for the isolations of new molecules. So I worked in this laboratory. He is the student of Asima Chatterjee. He is the first woman win the Bhatnagar Prize and first organic chemist, woman organic chemist, and first science PhD from India. So she also isolated vinco alkaloids. She developed anti-epileptic drugs and anti-malarial drugs. And we would like to continue this legacy and trying to support the natural product research. And my PhD guide always pushing me towards nutraceuticals and herbal medicine. You know the value, and he infused so much knowledge to Inatura, and he is advisor of our company for natural products. So this is again the regular schematic representation of natural resources. How we go for the isolation, maceration we'll do here. We do extraction, we'll go for the lead optimization. After lead optimization, after getting druggable candidate, which is, you can see some literature, IC50, MIC values, which are in micromolar or maybe nanomolar. So like that, after getting some druggable molecule, we'll go for the preclinical assessment and clinical assessment. Preclinical is for animal, and clinical is we'll try to check the druggability and safety, cytotoxicity of the drug application on the humans. That is clinical practice. Then we'll go for the initial drug approval, that is towards FDA. Then this process goes on, almost it's kind of 15 plus years of time and 1.5 billion expensive, highly expensive budget. So here is a I have already said that 40% of drugs are available in the market, which pharma drugs are natural products. So we'll try to optimize. Now we got an opportunity to highlight herbal medicine and natural product by using these AI tools and AI agents. So this is natural as are very complex structures, not like small molecule, how we design, what we design, as per our comfort, as per the bioavailability, we'll design and we'll try to check the, IC50 or maybe preclinical druggability, and we'll go for the market. This is easy actually. With the natural products, it's complex structures, but the application is very, highly applicable while going for any disease, treating. So that's where the complexity we can minimize. Here AI/ML we have to use to complex problems. We can simplify now. We have the tools. If you are not able to use these tools and agents, I don't think so you move forward in your career. Or maybe to highlight, or maybe you may not get valid data. Yeah. Here we can use for not only applications, one of the applications is target identification. Like we have to do market research or maybe literature search regarding any disease, anti-diabetic or maybe any kind of anti-cancer topic maybe you are towards working on lung cancer or breast cancer. So you have to check the market, which drugs are available, which natural products are available. So much, and you can check the data, and then you try to decide your target. How many drugs are available? Why our drug is suitable to, the natural product is suitable, and can we compete with this drug? So this type of things we can analyze by using AML tools and by visual screening. So mostly in visual screening, AMLs people are using, they are mostly going with the cloud. Whatever the AML tools and agents are cloud based. They got abundant data. Whole world, it is available related to what you are purchasing, what you are eating. This full data is available, and what you are taking, some medicine related and literature related, full data is available in cloud. So this cloud, this material they use, maybe like you are using ChatGPT, DeepSeek, or maybe Claude. So they recently what is Grok, right? These AI agents. So these are all cloud based. So then. Then we can check the biocompatibility and specificity towards the target. So here is the point is, I said 15 years time for the traditional drug discovery and trying to go with the market and get the success. You may not get success after 15 years also, but here we can minimize the time from 15 years to 7 years, from 5 years. Like during corona, we have to decide the druggable candidate and preclinical, clinical, and go to the market and giving to the patient within 1 year or 6 months. Everybody, like many people and many country stalwarts, they said simply to the scientific community, like big companies, to the AstraZeneca and Pfizer companies, within 6 months we need vaccine. So actually this is, at least for vaccine also it takes decades actually. But within 1 year we have to decide that time. We decided, and we made Covishield and Covaxin from Hyderabad Bharat Biotech. So here we can minimize. Now that time we don't have the AML tools, right? Now we have the AML tools. We can minimize the time to 5 to 7 years. So we can decrease the target identification time from 2 years to 6 months. And here hit discovery from 3 years to 1 year. So lead optimization, after getting the hit molecule, we'll try to, like, we can see lead optimization. I can give some example like that. If you've gone for interview, there's some levels, first level, second level, third level, fourth level. In first level, they will invite for 10 candidates. In second level, they will shortlist 4. In third level, be 2. Final is 1. Right? Like that lead optimization also we do. After getting some hits, we got some hits, 10 molecules, maybe 50 molecules. From that, we try to, from 10 molecules, we'll try to identify 4, which are highly potent and safety concerns, cytotoxics, everything we'll check. The latter, we'll try to move with the only 3 molecules or 2 molecules, while as a lead, the druggable candidate. After preclinical towards FDA and clinical phase 1, we'll go with only 1 molecule. So this is the lead optimization. We can decrease the time 25%, and we can do use for preclinical studies also AML, and for clinical studies also we can use AML. And genetic markers, we can biomarkers also highly applicable in the diagnostics, mostly diagnosing the corona or diagnosing any disease or maybe cancer, anything by using involved biomarkers. Whatever you do, flow cytometry, cytotoxicity studies, and maybe what we can say in vitro studies, you'll get some image, right? It has a biomarker already, like rhodamine. Rhodamine might is in the flow cytometry. We'll use that reagent, fluorescent reagents. So biomarkers are not very highly applicable. We can use for diagnostics, safety, and predictive pharmacokinetic, pharmacodynamics. I can use that one word. This is the applications of fluorescent probes. We are also already making the fluorescent probes, and this has diverse applications. These are biomarkers only. This is new term. Another alternate term. The sensors we can use for sensors, molecular probes, theranostic drug delivery system, and smart materials. Smart materials you can put like quantum dots. We have written some project for quantum dots actually. The 2024 Nobel Prize for quantum dots. I think you know that LED TV. Now the latest version is QLED, quantum LED. That is made by the China. And 2024 award Nobel Prize was the quantum dots only. I mean quantum dots, it's like a formulation. It's involved herbal actually, nano formulation. And some countries, that companies are using for roads, ships, making ships. While they use that some paint for the big ships. If you lost ship in any ship, you can see from the satellite after 10 years, 20, 100 years also. And after you made this road, and you can identify where we have supported some client or some project, we can identify by satellite after 10 years, 20 years. We'll add some quantum dots, which is like fluorescence only. It has this some little different mechanism, but it's very little we can add, and that is highly applicable in the future, near future. So here we made, you can see that anthracene. These are the structures which are, fused structures, mostly the fluorescent molecules. Anthracene, naphthalamide, pairin, BODP. I made in nuclear medicine department. I worked for 2 years in Samsung Medical Center with the BODPs. Those are asymmetrical substituted BODPs. We support for regularly the CT scan and MRI teams. Our technical team, they support, and we'll also support for cancer research also. And fluorescein and rhodamine. Right now we are working with the rhodamine actually, one project for a certain disease. We are trying to improve the fluorescence, and we are making some linkers actually. So cyanine, biotin, these are the examples. So here conventional medications also is applicable, the biomarkers, and we can choose from herbal, we can apply for herbal drug discovery. So here is the pros and cons related to biomarkers. It leads to personalized treatment, improved efficacy, enhanced safety. Cons is complex composition, product variability, lack of validation, inter-individual viability, ethical concerns. Like when we have to, if you want to tag some molecule and try to modify that molecule, definitely it would be some different molecule. So if some molecule is not having fluorescence, how it is leading, why it is leading, where it is interacted, we can see by tagging some fluorescent moiety. But after adding that fluorescent matter with the linker or some different mechanism, it's difficult to the bioavailability, and it would be a different molecule. Then there is a problem with the cytotoxicity, safety again. So there we have the complexity. Application ML in herbal drug discovery. That is, we use with the algorithms, conventional neural networks, so generative models. Right now generative models are ChatGPT only. Natural language processing and cloud. We are using cloud right now, working with the quantum AI towards anti-aging drug discovery. So these are some companies. Pfizer, they use from Fiverr, IBM Watson platform for immuno-oncology. From Sanofi, Excellentia AI platform for the drug discovery of metabolic diseases. and Genentech, GNS Healthcare AI platform for cancer drug discovery. These are software solutions companies only, but they are having the softwares to support the drug discovery. These are the mostly used platforms for the drug discovery. So here is smart molecules, one of the leading smart molecules right now, the PROTACs, proteolysis targeting chimeras. Like this area is emerging. Like you can see, that is green one is like one ligand that we are used to make with that one only one molecule to treat any drug till now. But here we made a linker and made some blue molecule, you can see, that is a CRBN ligand system. These are the big molecules. This is a molecule weight 1,000 above to 1500 kind of. The small molecules we use to halt some pathway, means simply mostly for the anti-cancer research. But you know, maybe cancers, you know, after cancer treatment, chemotherapy, many people are like, you know, their immune system is down, completely down. Maybe the treatment, after treatment, they may lose the life also. Most of the stories are not successful. But here we are not halting in certain pathway. We are not inhibiting. We are not trying to, you know, stop some time. We are here is this mechanism is trying to pull the complete bad protein. We have the bad protein and good protein. Bad protein is cancer. Good protein is normal cells, whatever we are having in the body. So this mechanism directly, completely take out the protein. It's not stopping. It's completely taking out the protein, bad protein. That's cancer tumor example. So here is a leading companies in PROTACs. They are invested billions of market is available, and many cancer centers, R&D centers are working on this platform. And if you would like to lead and trying to go with the drug discovery with the using natural products also, you can choose that area, and it's very highly applicable. You can get jobs. Career is, you know, bright. You know, recently from our team, on PhD candidate, he left to 10 days back and left for PROTACs team, South Korea, Yonsei University. So you can see Arvinas, Promega, and BioNTech, C4 Therapeutics. So many companies we are leading this area. So here we also working on PROTACs. We are supporting PROTACs R&D solutions. Here you can see these molecules are already, we have the building blocks molecules. We made many hundreds and thousands. That is E3 ligase system, which is CRBN VHL ligand. This is stable. These are stable, but the linker will make with the current library as per the client needs and as per the design of the PROTACs. Yeah. Just we use for the AI drug discovery DeepMind, AlphaFold for the biology. In silico medicine, you know, the in silico medicine company, they discovered one drug by using only virtual screening. It's not completely real time execution, like, you know, wet lab. We'll try to extract from some plant, some molecule. I mean, we'll make some molecule and library molecule 100 and 200,000, and we'll go for the screening. They didn't made any molecule here. So virtually they designed and they explored validated by using only software tools and using AML only. That in silico medicine company, you can check it in online. So yeah, applications. Finally, we have to conclude regarding AML, herbal drug discovery, target identification and validation. It very highly useful in identification validation also, compound screening and efficacy prediction and optimization, safety assessment, formulation optimization also we can do. Here is one of the work we are going through. It's 2 days back only we did actually. Yeah. So these molecules, like this is actually drug discovery towards anti-aging. We are not disclosing what is the PDB and whatever the details of that protein. But there is a green one is protein, that is inside the molecule is ligand. So this is simulations, MD simulations work. Right now we are working with the Quant IM team. Yeah, whatever the challenges, strategic solutions. And here is the data is very important. When without using AI also, using AI also, data is very important. Whatever the curated data we have, I don't think so. We sometimes there is a gaps and there is no original data. So there is a problems with the available data. According to data, if you give certain artificial intelligence tools and agents, they will give simply the conclude and they will give the result. But we have to check originality of the data available in cloud. How it is, for the scientific research. Different field is okay. For scientific research, we have to check the data is validated or data is really original. And regulatory approval. The regulatory approach is very critical with the classical approach also. But using AML is very critical. So regulatory people they have to change their rules and regulations to adopt these AML results. That is a roadblock. Yeah, strategic solutions. Yeah. So here is to collaboration is very key to excel in this field. Whether it is natural drug discovery or normal small molecule drug discovery, or maybe ADCs or different big molecules drug discovery, we have to collaborate with the software solutions team or AML team, as we are also have the software solutions complete AML team only. They don't know what is molecule, what is the chemistry. But we always keep in touch, and we got groups in WhatsApp and email communication. What is how is happening? We'll try to give the inputs. That is definitely, if you got the right collaboration, it's possible. Yeah. Yeah. Here is we have to check the models, the complexity, integration of the complexities is more while going with this field. But I don't think so. It's already happened. From 1 year, it's many agents are there. We are habituated. We can apply. We have to check the data quality, regulatory barrels, and we have to use many models. Which model is working? Because these models are been from 1 year only, not optimized. Yeah, herbal drug regulations, commercialization. How we can commercialize this drug using? We have to study the regulations. And we have to follow the GMP norms. So like we have to check if you are making some cosmetics, you have to check the cosmetic acts. If you are making fragrance, you have to check the fragrance act, how they made, what exactly they are made some rules regarding infrastructure check, maybe auditing. These things we have to check. To check that we have to be more professional with the QC and QA ability. So here is the new millennium Indian technology leadership. They implemented this one towards herbal medicine for the to change the regulations. So we have to support the projects by using this protocol. Yeah, we have to what we can say, for to produce any formulation or maybe an syrup or maybe kind of any cosmetic kind of any product, we have to take the Ayush license. These are really flexible, and they are comfortable with the startup ecosystem, MSME ecosystem. Yeah. So now the quantum computing is the era. Like quantum computing is like NVIDIA companies. Those are making chips. They are the highest gross and turnover. 4 trillion company, almost one country budget. So quantum computing we have to adapt, and real-time adaptive systems, whether it is matching with the real-time adaptive systems or not. Multi-omics programs. And we have to follow the personal medicine. Personal medicine word is not a new word, it's an old word only. But now we can apply. Yes. Yeah. Yeah. Here's our company, Inauguration Scientific. We do CRO, contract research organization. We support research and development related to drug discovery and development and natural product formulations also. So we started 5 years back, and we are supported for Singapore teams and Singapore and USA and Europe teams. Here is related to custom chemical synthesis, drug design. We support drug design and medicinal chemistry, custom synthesis scaffolds, impurities, intermediates we make, analogs, and API intermediates manufacturing, that is CDMO. We supported for some OMSI labs in Indore. Process R&D, that is pilot scale up, kg to multi-kg scale. Here we made some molecules, CL097, supported for US team, that is Covaxin adjuvant. You know Covaxin, right? This is adjuvant. We added they add for Covaxin. We support for some research team for USA. We supplied 1.7 grams. This is a CQ31 is a biochemical reagent. It is inflammasome. And the monobromobiamine, this is like a fluorescent molecule. This is we do fluorescent tagging for many proteins and many enzymes. That is pomalidomide, which is for Protax. So here is again like the Protax we support. These are the system, building blocks and libraries we got in-house, our inventory. Yes. Here we started 2-3 months back. We connected with some business development team in Europe. So those people suggested, and they after 2-3 meetings, they identified our potential. We supported the team members who are settled in Dubai. Like as a pharmacist, they are supporting for some German company, like air ambulance. They support many countries for the ICU management. And 2 people went to Dubai, and from one to USA, and recently South Korea. So they said like some people also get best employee awards. They are leading MNC companies. So we got an idea to send the students and support their research aspiration towards PhD program. PhD programs mostly completely funded. You'll get fellowship, monthly fellowship, not like postdoctorals, but you'll get minimal at least 1,000 USD minimum, I think so. We'll get the fellowship. During when I was working in South Korea 2011 to 14, that time also PhD students used to get the fellowship. So you'll get a fully funded PhD, MS plus PhD or MS programs. We got an initial discussions, and we got 19 universities agreements to support the manpower researchers from India. They are mostly interested in Indian students to take the abroad, offer the courses. So we also discussing which university is suitable for whom. I mean, maybe you may have to go with the IELTS. For MS PhD, I don't think it's not not required. But you need research publication or maybe research project, not only master's certificate or not only B.Pharm certificate. If you got a publication research project in your hand, you are attractive for them. So that's where we can fill the gap, and we'll try to support research projects. Whatever, if you got in some small project, we'll try to upgrade that project, try to train you in the wet lab. We know that in abroad what they expect. Their culture is different from our culture. Technically, I'm telling in professionally. Professionally, the culture would be different. When you enter in this laboratory, their rules and regulations are very critical. Yeah. So building the future with the one person is not possible. That's where we are happy to go with the skill development, collaborations, MOUs, and partnerships with the Indian ecosystem, scientific community, and academia and industry, and abroad also. Yeah, we must collaborate with the, just simply discuss with any AI scientist are there, like any AI employee is there. Try to discuss with them how these algorithms are working, how these tools are working, how these agents are working. Try to learn, and not only watching is using the chat GPT and using different AI tools is not enough. Go with some AI agents tools and get certifications. Try to get the job, or maybe try to go with the research center. Then definitely you will excel, and you will be placed well. Yeah. Here is like we have to remember that whatever the natural products hub as in India, we got the practice and we have the medicinal plants abundant, but we are not giving that much of support for the patients, maybe for many diseases for worldwide. So that is many complex issues, or regulatory issues, and maybe branding issues, or maybe we are not able to create that culture. But now the time came that AML is very applicable tool. If you properly use, definitely it's overtake the pharmaceutical industry and English medicine. For me, and as per our vision, we don't have any differentiation between English medicine and herbal medicine, or maybe natural. Medicine is medicine. Patient needs the medicine, some medicine to walk and go for the work, or maybe to enjoy the life, right? They don't want, they are not serious about English or maybe some herbal. Or they want, cure the disease and they want to walk. If you are in a bed, there is only one problem, right? No, there is no much problems. If you are healthy, you have many problems. You have to go for the, chill with the friends, or maybe go to movie, or go with the career. You have to build the house. But if you are diseased, there is only problem. No, I have to stand up and have to be healthy. So we are patient-centric. We are happy to support any type of strategies and solutions, whether it is herbal or natural product, or maybe English or some material, some using AML. So here is our company located in Hyderabad, near to Chellapalli station, recently installed. This is far from our unit, is far from only 1.5 kilometer from Chellapalli station. So yeah, thank you very much for your time and attention.
Thank you very much, sir. Anyone having any queries can ask now.
Good afternoon, everybody. Myself, Abid Hussain, and I'm coming from Nehru College of Pharmacy. And my question is to sir that how ready is India for adopting the AI and machine learning in drug discovery, either in allopathic or Ayurvedic systems? How ready is India regulatorily? Like, will they allow the use of AI or machine learning in drug discovery or not? In case of, we have seen in the FDA recently updates about the use of animals. They have replaced it with the AI chips and organic chips. They have used it. So they are replacing animals with the AI or chips that are being made recently. So how ready is India about this same thing? That's my question.
Hello, I'm audible, right? Yeah. So thing is, India is always ready actually, because we have the many software engineers, abroad, and we have the are Indians only. So it's possible we can adopt this AML, and if you are not able to adopt the AML, I don't think so we lead the pharmaceutical industry and whatever the herbal medicine, because of course I wonder that it's like been from many years we are services team for the pharma, we claim that Hyderabad is a pharma hub, but we are doing service. We are not providing solutions. But the branding products are for Hyderabad, USA only, USA and Europe. They do the final branding and solutions they will give, whatever the drugs approved and finally in the market and enjoying the patient by USA companies only. We are doing services through R&D and formulations, API, bulk drug. During Corona, had HCQ, one molecule is there, big molecule, a natural product type, that is when President Trump was also expected ton scale. He thanked for the Narendra Modi, Prime Minister Narendra Modi. So that time bulk drug ton scale we supported mostly almost 50% above. So but that is service only. That is drug is that product is we are not the owners of that product. but we are doing service. But if you use properly now, this is a time to change the culture. No, that's what I would like to say that don't study completely herbal medicine companies, herbal medicine, herbal research, and also compare and try to check some knowledge related to pharmaceuticals, English medicine, how these are growing, how these strategies made, how the brand is happening, how the regulators are approved, and compare this is the main competitor, no, English medicine. So that compare that their culture, how they are leading our main competitor, and try to infuse and try to implement in herbal medicine also, where we are missing, right? Trying to adapt the knowledge in herbal medicine only and trying to check the technical details, regulatory in herbal medicine, it won't work. We have to check the English medicine, how they are leading, and we can adapt. Here is a gap, this is a blank, right? What is, how, means like we can use AML. We have that chance now. So if you use, you can, it may definitely gives the solutions, how to lead. Yeah, I think I answered.
Thank you, sir. Thank you.
Anybody?
Anybody?
Thank you, sir. Mr. Tirupati Boini, Senior Scientist, Research Officer, Chemistry, specialized in phyto. He has been a part of multiple research projects focused on validating the efficacy and safety of traditional herbal formulation. His contributions included standardizing critical markers, AI tools for optimizing chromatographic methods. Having master's degree in chemistry, specialized training in instrumental techniques and NABL guidelines. Developed validated analytical methods for over 20 Ayurvedic formulations, actively involved in preparing NABL documentation and assessments. With immense pleasure, I invite Mr. Tirupati Boini, sir, for the section.
Once again to NCP and Nehru College Group of Institutions to today giving the opportunity to give this some topic, enhancing herbal drug discovery and safety profiling through AI-based phytochemical analysis today seminar. Okay, myself is Tirupati Boini, Research Officer Chemistry. I am working under CCRS. CCRS is like a Central Council for Research in Ayurvedic Science. Is the Ministry of Ayush. So I am working in Department of Quality Control in National Ayurveda Research Institute for Panchakarma. And today we are discussing with this topic. So I hope these are the topics to be covered in this session. So okay, those topics are like introduction of the herbal drug is safety, efficacy profiling, introductions to AI and search engines, what are the AI databases are available at present, and case studies on the herbal formulations through AI, what are the case studies are done in available in literature, and there are role of the AI tools in analytical method developments, the analytical chemistry, phytochemistry, and isolation of the markers in Ayurvedic formulations. So how that AI will be incorporated into our analytical method development, that topics also to be covered, and role of AI in quality control and quality assurance. If you are preparing any drug, any molecule that is treated your diseases, that should be follow the authority guidelines, Drug Cosmetic Act, those things are that purpose also. AI, how it will be implementing in this quality control and quality. So totally 5 topics to be covered in this my session. Okay, next The introduction to the herbal drug efficacy and safety profiling through phytochemical analysis. So before going that, this is what is the herbal drug. So everyone knows that herbal means plant. Those drugs are derived from the plant or medicinal drug. It is treat your diseases without harmful. Okay, very harmful also, very little bit, very less compared to the modern medicine. That's called herbal drugs. That most of the herbal drugs are either maybe single drug, either maybe multiple phytochemicals may be available. It's not only single phytochemical, most of the phytochemical may be available. It may be plant extract or isolation of the particular compound from the particular plant. So these are things that comes under herbal drugs. So most of the herbal drugs are widely used in Ayurveda and traditional medicine systems and Unani and Siddha. These are the traditional anesthetics that people are using this herbal drug industry. Okay, what are the characteristics are the herbal drugs? So why we need to study? So what should be the characteristic? Why it is called herbal drugs? So those are the major characteristics. They're derived from the natural source, and they should contain maybe single herbal, polyherbal formulation. If you take Chyawanprasha, maybe 30 to 40 medicinal plant will be there. Okay, if you take Ashwagandha, only single plant. If you take Piper nigrum, Piper churna, like piperine churna, only single compound, but it's having so many pipernoids. If you take curcumin, curcumin longa is like a single herb, but it's having curcuminoids and 2-3 more. So such a way, it contains an active phytoconstant. That active phytoconstant may be interact with your enzymes or amino acids, and there will interaction, they will cure the diseases. So this simply is nothing but chemistry is there. That chemistry may be interact with your enzymes and cure the diseases. So that's available also. Those herbal drug medicines are like maybe powder, maybe decoction and extracts and capsules, oil and ointment. These are the comes under the herbal drugs. Okay, and simply if you can do the pictorial representation, same herbal medicine, the different of formulations, extract, granules, tablets, capsules and emulsions, and maybe those like if you have every plant having some total phenolic and total flavonoid, that total phenol and flavonoid interact with the silver and gold, maybe making nano emulsion. That nano emulsion also may have used as a nowadays in herbo mineral medicine for the cure the anti-cancer diseases also. Okay, these are the others comes under the herbal medicine. This light introduction. And next, most of the herbal plants are considered plant based only. I am saying that root, stem bark and fruits and seeds. These are comes under. So we are the examples are giving Ashwagandha. What is the botanical name? What is the part used? What is the therapeutic effect of those? Okay, these slides will be shared to you. You can take it from the madams. Okay, these are the plants. Okay, what are the scientific name? What are the leaf? What purpose they are using? Already the existence in Aniscent and API, Ayurveda Pharmacopoeia India also is mentioned. But those things we will call as a herbal drugs. Okay, like Gugulu, Camphora vitae, resin we are using, that cholesterol control, arthritis, and amla, Indian gooseberry, fruits are using antioxidant and other anti-aging property, and Brahmi, Bacopa murena, old plant, memory enhancer. These are the herbal drugs plants are using. And what is the herbal drug safety and efficacy? Why we need to study the safety and efficacy? What is the safety? If you are going traveling to bike, you should wear safety. Safety is first. Safety is first, you can say, because you should wear the helmet. Such a way, if you are giving a drug that any human kind of society, that should not much more harmful to the human beings and whoever take that herbal medicine. So the safety is matter. So nothing but safety is the herbal drug safety efficacy is evaluation and assurance. Evaluation means the works to indent. So whatever the drug is taken, the drug should be effectively cure your diseases. It's not harmful to the much more as modern medicines are doing. So these are called the definition of safety and efficacy of this. These are the regulatory approvals should be, like if you are preparing any formulation, this should be safety efficacy is mandatory. Without safety efficacy, your drug will not come to the market. You cannot patent it. So the most of the herbal drugs are the phytochemicals are used. The phytochemicals is for therapeutic activity. And if you go for this, what is safety concern? So we need to do the this herbal drug. What are the evaluations methods? These are the safety concern, toxicity. So it may be hepatotoxicity or nephrotoxicity. Hepatotoxicity means it will damage the liver or not. That study should be done. And the contaminations, because in most of the herbals we are taking from the field and out road, sometimes nearby roadside or some of the industrial area side we will collect it. But they're containing having heavy metals, pesticides and microbes. So as per Ayurveda Pharmacopoeia India and CCRS guidelines, what are the heavy metals we should focus it? What are the limitations for that? So as per Ayurveda API, the heavy metals are 4 things we need to study. that is like lead, cadmium, mercury, and arsenic. Lead is 10 ppm, arsenic is 3 ppm, cadmium is 0.3 ppm, and mercury is only 1 ppm. Such a way some limitations is there. Okay, those things also we need to study. And adulteration, the addition of steroids and synthetic drugs. You know that sometimes what's happening, if you pure curcuma longa we can take, sometimes that yellow color dye also will be mixed with that curcumin powder. Because if you purchase from the natural resource, that curcumin content is different. If you are purchasing from market curcumin powder, that is maybe different. So some people will be do the adulterant, because this agency very smart as compared to we. So they will add some dyes also. Those dyes are, we need to check it out. That safety concern will matter. And next, drug herbo interaction will be important, and dosage and self-medication. So self-medication also will be safety concern. Dosage also, if you take paracetamol 200 mg is sufficient for child. If you give 600 mg, it will be finish your liver. So those dosage errors also will be safety and concern. So what are the methods to evaluate these things? These are the things: preclinical toxicity testing, and that may be acute and subacute and chronic toxic animals, and in vivo, in vitro, and next is toxical screening, AMS test, and LED50, and clinical safety, trial 1, trial 2, and pharmacovigilance. So these are the safety concerns. These are the evaluation methods to the know the drug is safety or efficacy. Okay, next we will go for the, everything is phytochemistry, or as phytochemistry point of view, every plant having phytochemicals. So what are the phytochemicals may be available? The major phytochemicals are like these only: alkaloids, flavonoids, terpenes, glycosides, safenins, and tannins, and polyphenols. So if you go for the alkaloid, examples are morphine, berberine, it potent pharmacological actions, and quercetin, camphorol component, flavonoids, antioxidant and anti-inflammatory, and terpenes like curcumin, anti-malarial drug. So if you know particular marker in your herbal, you can do it this study. If you don't know, we are the new like plants you are getting that is having some activity, you can do the total phenolic and total flavonoid. Total phenolic, which self to gallic acid as a standard, you can check it out. Maybe gallic acid and their derivatives may be available. The total phenolic count will give that my plant is having, my extract is having this much of gallic acid, it will equivalent to total phenol. So it equivalent to gallic acid, we can say. And flavonoid purpose, camphorol or quercetin also we can take as a reference. Okay, this study we can study those things. Next one is, so what is the role of this phytochemical? Means that every phytochemical having some therapeutic role. That example where it interact with receptor, enzymes, and cell signaling and pathways, and multiple target action with the lower side effects. So, and compared to modern particular single herb, this is multiple phytoconstant may be there. Some will be activator energy, some will be curing the diseases. So less side effects will be there. So why we need to identify the bioactive marker? Means by, you see, every plant having more than 2 phytoconstant, but every phytoconstant may not be therapeutic effect. Because if you take like vithanolides, vithanol A to D, and other than some other phytoconstants also will be there. Vithanolides A to D the therapeutic effect, but qualitative purpose other phytoconstant may be check it out. So those are effective for the particular disease, that is called bioactive marker. This bioactive marker we need to study, and these are the examples of 2 chemical compounds. Maybe first one is quercetin, and this is morphine. These are the phytochemicals we need to study. Nothing but the phytochemical interact with your enzymes and cells and receptors. It will be interaction will be there, then they cure the diseases. This is a basic fundamental. So what are the challenges are there in the safety and efficacy in herbal medicine? Those challenges are like this: lack of systematic toxicity data, and that limited edimate means absorption, distribution, metabolism, extraction, and toxicity. Absorption means how that molecule are given to the particular person, how it will be absorbed your blood serum, and distribution means how it will be distributed to body fluids, and metabolism means how it will be affect to the livers and kidney, and extraction means how it will extract through the urine, and toxicity is how the acute toxicity or normal toxicity, these things will be lacuna, and batch to batch inconsistency. Like if you are taking any ashwagandha churna, that plant is collected from the Tamil Nadu, same next batch we are collecting from the Kerala, other third batch will come northeast. So that selection of raw drug from different resources, so it having different content of vithanolides, because if you are taking one plant, you are growing in proper condition and proper climate, other plant you are growing in like drought, like in like summer season, that having phytoconstant will be different. So such a way the batch to batch constants also will be differ if you are taking a raw drug at what are the medicine from different locations. So those batch to batch constants are having lacuna, there it will be like these are some of the pictorial representation. These are challenges of traditional formulation: authenticity and accessibility, standardization, regulatory authorities, and validation, efficacy and safety to convert to a nano gels and different formulations and modern medicine. These are the challenges, and this is a pictorial representation. These are the I say now, variability of plant resources. Example for the soil and climate, season, harvesting time. So vithanolides are 2 to 6 variations are varying with the vithanolides content across the region. And other one is adulteration. Some of the Astrolochia species having the ANN properties. And other one is incorrect dosage and lack of standardization, overdoses. These things will be contaminated. And lead contamination is 20% are available in market available Ayurveda formulation. That is harmful. As per Ayurveda, 10 ppm is sufficient, but is some of the medicines having more lead contamination. These are the safety. Example of we are taking in one ashwagandha. Ashwagandha vithanolides will be differ from the climate and forced harvesting time, and it will be differ for different resources as we said. And these are the example. I will take it here. Same ashwagandha from the different sample code but different region, that vithanolide components of third column it will be differ. So the particular content of vithanolides are safe in safety risk, but more than what we can say 5.5% of vithanolides will be heavy risk, and it will be damage to the some particular organ. So these things also we can need to study the drug prepared formulation having what are the content are there, what are the limit of that phyto constant. So these things we will study. So how we can study these things with help of some modern techniques. Those are what? Like HPLC instruments we can use it, and we can study that. These are good, but they are having limited to human opportunities. Everyone we cannot go for the to analyze their particular marker, particular scalable, and thousands of compounds we cannot do it. These are the HPTLC, ChemAg and HPLC, SHIMATS, and here it will come to the artificial intelligence. So how artificial intelligence will help to the here, because it is artificial intelligence, it collect all the literature, it will be processed in large databases, what are the phytochemicals already existed in particular plant and particular extract, and how it will stimulate the interaction human targets, and edimity properties like absorption, distribution, metabolism, excretion, toxicity. These things will be give the idea of the AI. So this can be the possible pattern and revolution to the herbal pharmacy. This year it will come to the AI. So now introduction to the AI. So search engines and phytochemical analysis. So simply, what is AI? So as a chemist I will say maybe aluminum molecular formula is AI, like AL only. It will look like aluminum. So it's not aluminum, it's like artificial intelligence in this today's seminar. Okay, we can say artificial intelligence nothing but what is artificial intelligence? It's like stimulations of human intelligence in missions. What are the human beings are thinking, same such a way the mission will be think, they will gather the data and it will give. But it's not 90% accuracy, maybe we can believe it. And again we can go through the human beings. So machine learning and deep learning things also will be there. These are the herbal traditional formulations. It all gather data together and it will analyze, it will give these are maybe solution. Okay, so AI means not aluminum, artificial intelligence. Okay, these are the artificial intelligence, what it will be help, how it will be support to the as a researcher, or it will be literature review it will be done, and it will be the phytochemicals data it will be provide, and the method development for the keynotes for the new method development or sophisticated instrument like HPLC, HPTLC, GCMS, and ADMET properties. This is much more important for the if you are drug preparing clinical, preclinical, and quality and safety, and bridging ancient wisdom to the modern data science. So these things will be done the AI. Okay, why AI in phytochemicals? As we say, lot of phytochemicals are there. If vithanolides and ashwagandha having so many vithanolides, sir, which vithanolides is especially cure for particular diseases? So those data also it can be given for the AI. Okay. Next, what are the key applications and what are the examples of herbal research area used in QSR? Are also predict the activity and toxicity. Visual screening will be happen. Curcuma analogous also using deep chem models. It will be how it will be useful. Those things are already given here. These are the tools, machine learning tools, and random forest, SVM, XBoost are the active and inactive phytochemicals. It will be given for this database. And example predicting the antioxidant property of polyphenols. These are one of the paper 2021 they published by using these machine learning models. And deep learning models also like neural networks and CNN networks, and these are there already they used. And QSR, qualitative structure research activity leadership models, are used to the hepatotoxicity of alkaloids and what are the lethal dose and what are the HERG inhibitions already published in this paper. I can search it. What are the tools and platforms are available in this checking and everything. One is AutoDock, molecular docking and interaction analysis will be done. Second one is Deep Chem, deep learning of drug discovery, and NIMI, Orange, Chem Profile, Chem Probe. This is property of prediction of using graph neural network. These are the online available, some of the payable, some of the open access things will be available in network. So these are the one of the database. I will give the phytochemical database. This is IMPACT, Indian Medicinal Plant Phytochemistry and Therapeutics. Another one is NPOS, Natural Product Activity and Species Research Database. This phytochemicals, if you want any particular phytochemical, you can give the phytochemical name, that what are the plants will be source for that phytochemical, it will be given, what are the activities done. So everything will be given by this database. IMPACT database is built by the IMS, Institution Mathematical Science for the Chennai. Okay, they prepared. This is also AI based network. It's provide 2D and 3D chemical structures and additive properties. That molecule, it will be much more interact with which enzyme or which amino acid, those things that also will provide this adimate. Other than this, these are the that IMPACT database will look like. It will give, if you take piper beetle on a plant, what is the kingdom, group, synonyms, what are the linkage, the smiles, interactions, with what are the enzymes, this database also will be provided. So another one is NPAS. NPAS is natural product activity and resource done. Most of the time natural products are isolated from the plants only, that 50% of natural drugs are approval. So there it will go for same, this database also will provide the physiochemical and chemical properties, how the molecules, molecular weight and hydrogen bonding donor and hydrogen bonding acceptor, like source of organism, where the plant is collected, when is collected, what is the harvesting time, what is the location time, like references, everything will be given. This database, natural product reference database. Okay, these are the natural product source. It is started in 2017. It will be collecting all the data and centralize it to the database. These are the activity studies are already done. These few papers we can get it. And these are the future extraction. There it will be give some smiles. The smiles may be utilized for the protein and ligand protein interaction time, and these are the one examples. So what are the data we will give, how it will become, these are the values. And next, these are the entire output which will come like this. If you take any phytocompounds, they predict the safety and activity, and these are compounds, curcumin, bis-methoxycurcumin, astrolic acid, what is the bioactivity score, what is the toxicity, is low or moderate, and the ADMET grade also it will give. So almost the ADMET A grade will be good. These are the chemical structure of curcumin and methoxycurcumin, astrolic acid. So these type of output data you will get it. So I am giving you only very brief introduction of what type of data you will get it. Okay. Next, if you go for the TKDL, this is very old one. The CSR institutes are providing this traditional knowledge digital library database. This contains almost 4.5 lakhs formulations and practitioners, and it's available. Those Ayurveda, Siddha, Unani, Soyaripa, Yoga people are using what are the formulations and drugs, and it's also available in 5 languages: English, Japanese, French, and German, Spanish. Okay, this database also we can utilize it, because what are the drug you are preparing, they definitely will ask what is the resource. So what is the reference for that? Already this is proven in ancient or somewhere it will be proven or what, such a way they will ask. So we can use this TKDL also. And next one, same TKDL having number of texts are included for transcription. Ayurveda is 119 and Siddha is 91, Soyaripa is 1, total 281 textbooks are they prepared, they are doing transcription for the digital. And now recently AI, that TKDL also providing AI based traditional software that recently 2 days before their launch, and it's not came to the online. So this is also TKDL give the India first, India is the first country to world digitalize the traditional knowledge to the digital mode. So this is also started. It's very good initiative for the government of India and to providing the traditional knowledge to the digital platform. And next one is case studies. Okay, these are the one of the case study like anti-cancer herbal drug. They are screening the flavonoids and AI-based QSR model. They get the model accuracy is greater than 90% 10-fold CV, and they reduce the experimental value is 60%. Another case study, AI in the herbal drug, and this paper is published in Phytomedicine 2021. Here the 3 different formulations are taken, the ashwagandha, gugulu, and QSR, triphala, there also accuracy is more than 88% and predict the, these are the activities. And another one is prediction of the hepatotoxicity. Here also 10,000 plus polyherbal formulations are randomized forest model they are used, and their toxicity also they study the LD50 and ortholic acid and piperine high doses is heavily harmful to the high risk. And another case study is like this entire the 3 case studies like different focus of study and what are the herbal formulation they are used, what are the techniques they are used, and what is the outcome is came, what are the references. These are like that they are using AI based softwares and study their acuity studies. Another one is the role of tool in chromatographic analysis. So that is like different diseases. But I am as a like analytical chemist, so how it will be used that AI in my domain. So it will be how it will be. So what are the challenges are I am facing? Those are the benefits you should be give the AI. So what are the benefits for the AI in method development? So if anyone you knows the HPLC, what problem you people will face? HPLC, what problem you will face? Most of the time solvent, sir, much more solvents are there. Which solvent I want to take? Sir, so many columns are there. One column will be 150 mm, one column is 250 mm, someone will be C18, someone will be C18. What is that C, what is that 18? We don't know. So how we can inform to the analyst? So my compound is having particular marker. So those times it will be benefits of this AI in method development, because save the experimental runs and solvents 40 to 50% reduction, and improve the reproducibility and standardization, and enhance the marker identification, and enhance the accuracy and precision is matter when your method development is going on, and optimization experimental condition. So what is the temperature I need to make it, what is the runtime I need to make it, what is the flow rate I need to make it, these things will be useful, and these are the things. So AI assisted already in HPLC, HPTLC, the people already studied, they are using genetic algorithm and artificial neural network in optimizing of retention time. So those berberine from HPLC, reduction time is mean average error is 0.2 minutes only. But prediction and experimental values are observed, and prediction and experimental values are one more case study here. Like camphorol is predicted 0.78 and observed is 0.76. So those data also it will come. These are the software tools they are used in papers, MATLAB and QSSR model and deep learning retention organization. These are the databases, payment bases are there, some of the fee are also available. So these are the like entire few references I took from the literature existence. These are the phytochemical, there is a source, and what is the AI they are used, what is the predicted RT and RF, what they are observed. So almost all are 0.1 to 2 variation only they observe. So they will give this, save the much more time. Okay, these are like few more example like quercetin and gallic acid, beseline, vagnine, and another one is like picoside 1, eugenol, alpha cyprin, desazine, plumbagine. These times it will be the already they done by using AI usage, the prediction and observed RF values. Other than this, these are the another few more things, and AI tools and what are the resources they are utilizing. These are the tools and their application, their websites. Okay, we can take it like a screenshot and slides also will be provided. We can use it. Some few are the like payment basis, few are the subscription basis are available. These are the data. Okay, next in the, what are the troubleshooting you are facing in AI in Herbal? Is it like what I said now? Most of the time with analytical, we can use HPLC, GC. There's some of the problems, the peak overlapping, retention time, baseline noise, and these things will be common method development challenges, troubleshooting. These are the one of the case study, and AI assisted pH and mobile phase optimized by berberine from the Berberia arista. And this is a result. Another one is like one more role. So majorly is like historical data is will be collected, and pattern recognition will be also, it will give the, and predicted analysis and mobile phase, and what is the, if you run the HPLC, it will be isocritic is good or gradient it will be good, that will also will suggest the AI in troubleshooting. And these are the tools they are using chromatographic troubleshooting. These are the tools and purpose, that are the website platform. Okay, other than this, this real-time monitoring also it will be done. Okay, if you having any query like overlapping FTI spectra, some polyherbal formulations, how will it will be solution? The PCA, principal component analysis, and SVM isolate key, the it will be separated out how the peak will be overlapping, which method is good, what condition is good, it will be provide. If unknown peak will be coming in UV visible spectra, based on that AI database it will there, maybe it will be polyphenol, maybe it will be camphorol, it will be terpenoid. Those data also will provide in troubleshooting. And other than this, these are the key benefits we will take in AI from troubleshooting, like time cost saving and high reproducibility and improved accuracy, automatic calibrations and system maintenance. These are things will be done. And next one, there are key takeaways from the AI. And the drastically human trial errors will be there, that will be reduced, and provide real-time automatic diagnosis and complex matrix handling easily. And other than this, these are the same websites. And simply, if you are like if you're ChatGPT are using now, that where we want to go particular matrix purpose, we will check it out ChatGPT. Same way you can ask ChatGPT also. These are the reverse phase HPLC method. These are the mobile phase I am using. These are the like phytocondition, maybe where it will be come to the RT. So it will give you some idea, because if you are giving some person to HPLC person, you are I want to run my medicine in HPLC chromatogram, we need to tell how much time I need to run. So if you are, what are the phytoconstants available in your formulation, so you need to know which time it will be elevate. So it will give some idea, it will provide it in ChatGPT. So these are the, my phytochemicals are present in particular formulations. These are the run time. So these are the RT will be provided, though I will say that don't run for 30 minutes. If run only for 15 minutes, that every phytochemicals will come out in a particular mobile phase. And also it will suggest AI, like ChatGPT, don't go for the these mobile phase. Maybe some of the peaks are not evaluated properly, not so making suggest alternative also, it will suggest. It will be time saving and money saving. Okay, these are the one of the other case study HPLC. This artificial intelligence they are using this. These are the predicted value and experimental value with the two analytical techniques, like that's ANN and REG. And this is one more ascorbic acid and paracetamol combination. Another one is these two things are there used. So here that the conclusion is that in this study they are getting very good results in the REG, multiple recognition analysis, and they are getting also mean square errors and average square errors, and the predicted experimental values are very clear. And that two software they are used, and that that neural network AN method is much more better the predicted value than the regression modes. The REG they are used, and AN also used for this combination of the chemicals. But there that ANN model is very good for this their particular complex. Okay, these are the like the tabular column, what is the correlation effect and coefficient relation. Those things are very similar. And these are one more techniques like liquid chromatography technique they are used in lab experts. This is like atomized computerized development system. It will give the real time and artificial intelligence system which method is good, and is isocratic is good or gradient is good. So what are the method we need to check it out, such a way it will go. It will give the like if you go for the gradient run, it will come this much time. If you go for isocratic, this much time it will be available. Such a way platform already available and that lab expert. And here also like they are studying injection in gradient and injection is isocratic. If you go for the gradient, it will become like much more peaks will be clumsy and overlap. If you go for the isocratic, the peaks will be differ. Which mobile phase is sufficient? There also 80-20 water and ACN, here like 50-50 ACN and water, that isocratic way. Well, isocratic way is much more better than this gradient. Such a way also software is already inbuilt in few HPLC system, that is also worked on AI based only. The finally C18 column injection in isocratic, and what with 60 and 31 is better separation for the particular 4 components. Okay, these are the one of the paper study, and these will databases also it will be provided for the validation QA and QC purpose. These as a chemist in my lab department, we published on paper in the base of this randomized response surface methodology by using design expert software. So we have the embryonic drives when we will isolate the embryolin. So I need to check it out. Solvent I fixed, but how much time, how much temperature is required to extract the much more embryolin from the embryonic drives. So if I want to do the normally, I need to do work it 1 month. So before that 1 month time, I don't need to waste. So I given the software, and these are the like commandings I given. Code 1 like temperature is 15 to 40. Like temperature is 30 to 80, minimum is 30, maximum is 80, and like time is like minimum time is 15 and maximum time is 40. So entire this data, the 3 factors are responsive, and it will provide the data is like this, some graphical representation, and entirely what are the like F value, P value, all are coming under the as per ICH guidelines as less than 1. And these are the runs, 17 runs already done in software. It provided the prediction and actual values, how much time is good, and yielding us what is that, those things also is provided. So after extraction of those molecule is same or not, I went for the HPTLC, the different wavelength. So first one is like standard, second one is I observed crystal, the next three are the chloroform extract. So I did everything in chloroform extract. After that, the obtained molecule I analyzed it through NMR. So those are the embylin, like the different carbon hydrogens are observed in the NMR chromatogram. So I find out. So at the final conclusion of the software, I optimized. So one part of drug is required, 3 part of solvent, that is chloroform, and 34 minutes and 49 degrees centigrade is maximum extraction of that particular isolate compound. Okay, this paper is published in JAMFAS and last 2 years back. So this type of, like if you're doing any extraction, isolation of products, we can use for this software. Okay. And next one is like one of the in silico studies also we did in corona time, like corona, that AC incubators, where most of the time the people are using I-64, some Punarnava, Quatachurna. Punarnava is like Borevirian diffuser. Borevirian diffuser, so many phytoconstituents. On that phytoconstituent we selected beta carotene and litronin, retinol, and these things. Here that based on this interaction with the different ligands, there's what you can TAPCA and number of hydrogen bonds and log P values. Based on that, it's decided that litronin is giving best components to cure the ACE2 inhibitors and COVID-19. These are the interaction stimulations of the software. SysMade we are using in online source available free software. So finally, like these are the amino acids. So these are the phytoconstituents. What are the amino acids interactions are there? What is their bioactivity score? And how it will be pathway to the channel. These things are, it obey the Lipinski rule and good affinity and AC inhibitor to come back to COVID-19. So these also published in Natural Product Science, one of the paper. And these are what are the role in quality control assurance means. Simply it will be image processing and HPLC image processing also will be done if you're providing databases and spectroscopy and chromatography. Okay. And next one is evidence of these things is machine learning, or you are using deep learning also we can use it, different purpose. And another this automated fingerprinting profiling and AI based classifications and image recognitions, chromatographic modeling. These are things we can use it in adulterations detection from the with help of AI. And other than this, impact of quality assurance and consumer safety, it will give the purity and safety, either it will be safe or not. If having pure compound, only one chromatography, one peak will come. If have another extra peak will come, this may not related to your compound. Such a way also purity and safety and trust and integrity, ethical sourcing and standardization. So this is which way it will be impact and quality assurance and consumer safety. And what are the challenges are and limitations of AI means that AI nothing but whatever data is available in cloud and research, they combine together and it give it. If you giving data is wrongly, you don't know AI, because if you one child you are giving one chaku like one talwar, he don't know how it is utilized. As cutting of vegetables or cutting of someone. So such a way AI means we should provide ethical is mandatory, regulatory aspects and interpretability, where the data is collecting from is good data source, database or not, some unknown person providing database also is mandatory in the quality control. These are the references I took from the gathering of these presentations, and these are I very thankful to the NCP and Nehru College of institutions, group of institutions, and also thankful to the Director General CCRS giving the opportunity working as a research officer, the council, and also thankful to my director and NIRP family members. I am also thankful to one and all your valuable time to listen this much of topic, and also thankful to my teammates. These are the botanists. What are the plant will come? The plant should be, as we said, Nandakumar said, the plant should be authenticated to be identified proper or not. So Tulsi will be botanist, and she will be identified the particular species. And next Rahul Maurya is the pharmaceutics. He also did that in silico studies and isolation of extraction, that QBD model. Lakshmanarayana Mishra, HPLC expert, is the developed analytical method development. Myself is only gathering all the data just in front of you and telling. Okay, these data all done by these well-wishers and my supporting my teammates. And thank you one and all. If you have any query, you can ask.
Any query?
There.
Good afternoon, sir. Myself, Abid Hussain, and I'm a final year student coming from Al-Shifa College of Pharmacy. As we saw that AI and manual reports, they were almost accurate. Not that accurate, like precision was there to be seen. Are we ready to fully depend, like can we take the reports only from AI? Like we are doing the manual observation as well as the AI. Which time or when we are going to fully rely on AI? Are we capable of that? Should we trust AI fully in that case? Because quality control, we all of know that quality control in drug production, drug development is a crucial part.
Yes, yes.
So are we going to fully like apply or fully accept the AI, what is it giving, or we should go parallel along with the manual and AI, that's it.
So one simple example I will tell you, like don't take it in personal way. One case is happened in Uttar Pradesh, somewhere in north side. The people are taking Google map, they ride car very speedily, that bridge is broken and they died. So because that AI is like giving the opportunity, saving our time and comparable data. Don't believe that 99% and like 100% AI. So it is a tool for the supporting data. Like as a guide or a teacher, it will be support you. So don't take it 100%. AI is coming. See what are the I given that picroside 1 or 2 RT. But there also I will get it changes in this mobile faces and as well as RT. So don't believe 100% AI. Simultaneously you can use it. Okay, that part, as a part you can use it, because if you are, nowadays is everyone having smartphones and ChatGPT, everything. So if you're going library and going the book references, it will be the idea. So based on that, you can go for the experimentals. So don't believe 100% on AI. AI is good, but don't believe on that.
Thank you, sir.
Okay. Any other?
Sir, can you suggest any one analytical tool which is used for the analysis of herbal medicine, sir, in AI? AI-based analytical tools.
AI-based analyticals, so many databases are there. Now again, like AutoTac and ChemoProof, and those things also like not freely open. It having subscription basis. There, if you provide your chromatogram, like HPTLC chromatogram is there. If you don't know everything, just provide it. They ask some questions. What solvent are used? What mobile phase are used? What plant you are used? So you can give those 4 parameters. It will give that these are the RF may be related these compounds. Like it will give simply, it may be flavonoids, it may be terpenoids, it may be alkaloids. It's not give exact. Like if you run the any phytochemical in GCMSMS or LCMSM, it gives the probability. The based on the library, NIST, such a way it will give it. With already LCMSMS already is provide itself is software built, and GCMSS also it will provide it. But HPTLC, HPLC is not developed, but few softwares are there. If you go at the chromatogram, we need to give these 4 data. What mobile phase you are using, what plant, or what type of solvent you are using, those things you can provide now, it will give this rough idea. Probability it will give. Already LCMSMS also giving you probability. Those probability more than 60% we can authenticate it. Okay, those we also itself software itself already. If waters itself having Shimarju also itself you having LCMSMS also it will provide.
Sir, is it only available for industrial purpose, or it is available for organization for institution purpose also?
Because those who are using LCMSMS, you can send it sample to them. They will analyze and they will give it. They give it liquid chromatographic chromatogram and what are the probability of compounds. They will provide. Those who are sending sample to LCMSMS analysis, like IIT Calicut also having, not Calicut, sorry, Palakkad. IIT Palakkad also having LCMSMS facility. IIT Madras also having. IIT Bombay also is having. Those you can send it. And our CSMDRI Chennai also having GCMSMS facility. You can send your formulation, ask them what are the probability of compounds. It will give it and give it. Any other?
Thank you, sir.
I hope now we can say what is AI, not artificial intelligence. Okay.
Okay, thank you, sir, for the informative session. So already later in area, it's time for lunch break. Appo first, we have poster presentation. Poster presentation ന് പാർട്ടിസിപ്പൻസ് ഉണ്ടെങ്കിൽ അവർ ആദ്യം പുറമേ നമുക്ക് ബസ് അറേഞ്ച് ചെയ്തിട്ടുണ്ട് നാളെ ക്യാൻറ്റീനിലേക്ക്. അപ്പോൾ പോസ്റ്റർ പ്രസൻറ്റേഷന് പാർട്ടിസിപ്പൻസ് ഉണ്ടെങ്കിൽ അവർ ആദ്യം ഫുഡ് കഴിക്കുക. ഫുഡ് കഴിച്ചു കഴിയുമ്പോൾ ക്യാൻറ്റീൻ്റെ പുറത്ത് തന്നെ നിങ്ങൾക്ക് കോളേജ് ബസ് ഉണ്ടായിരിക്കും. കുറച്ച് പോസ്റ്റർ പ്രസൻറ്റേഷൻ്റെ വെന്യൂ അനുസരിച്ച് നമുക്ക് കോളേജിലാണ് പോസ്റ്റേഴ്സ് പ്രസൻറ്റ് ചെയ്യുന്നത്. അപ്പോൾ ക്യാൻറ്റീൻ്റെ പുറത്ത് തന്നെ എന്തുണ്ടായിരിക്കും, ബസ് ഉണ്ടായിരിക്കും. അപ്പം നിങ്ങൾ അങ്ങോട്ട് പോസ്റ്റർ പ്രസൻറ്റേഷനുള്ള പാർട്ടിസിപ്പൻസ് മാത്രം എന്ത് ചെയ്യുക, കോളേജിലേക്ക് ബസ് യൂസ് ചെയ്ത് പോവുക. റിമൈനിങ് ഉള്ളവർ തിരിച്ച് നമ്മുടെ ഈ ഓഡിറ്റോറിയത്തിൽ വന്ന് തന്നെ അസംബിൾ ചെയ്യുക. നമുക്കൊരു 3 o'clock വാലഡിക്ടറി ഫംഗ്ഷൻ ഉണ്ടായിരിക്കും. അപ്പോൾ ഇപ്പോൾ നിങ്ങൾ പുറത്ത് നമുക്ക് ട്രാൻസ്പോർട്ടേഷൻ ഉണ്ട്, ക്യാൻറ്റീനിൽ പോകുന്നു, ലഞ്ച് കഴിക്കുന്നു. പോസ്റ്റർ പ്രസൻറ്റേഷൻ ഉള്ളവർ ഫസ്റ്റ് ലഞ്ച് കഴിച്ചതിന് ശേഷം കോളേജിലേക്ക് പോവുക. അവിടെ പുറത്ത് കോളേജ് ബസ് ഉണ്ടായിരിക്കും, സ്റ്റുഡൻറ് വോളണ്ടിയേഴ്സ് ഉണ്ടായിരിക്കും. തിരിച്ച് ബാലൻസ് റിമൈനിങ് ഉള്ളവർ ഇങ്ങോട്ട് വരിക. നമുക്ക് 3 o'clock വാലഡിക്ടറി ഫംഗ്ഷൻ. ഓക്കെ, താങ്ക് യു.
plant is collected and it is identified, then authenticated by a botanist and costly dried and utilized for pharmacognostic and phytochemical studies, in which pharmacognostic studies include macroscopic, microscopy and powder microscopic evaluation. Then phytochemical studies, which involves the identification of active constituents. The research findings are: in macroscopic evaluation, we evaluated the color, odor, texture and taste of the plant. And in microscopic evaluation, the transverse section of Scavilla taccata leaves is shown, in which it contains barrel-shaped vascular bundles, collenchyma, and prismatic crystals of calcium oxalate. Then powder microscopy also reveals that it contains xylem vessel, phloem, epidermis, mesophyll, calcium oxalate crystals. Then into the phytochemical studies, determination of physicochemical constituents, it contains moisture content, ash value, extractive value, which all falls under the limit. And this drug is successively extracted using petroleum ether, chloroform and ethanol, in which ethanolic extract gives more extraction. Then these extractive values are determined for the preliminary phytochemical screening, in which petroleum ether fraction have terpenoid content, where chloroform fraction have terpenoid, flavonoid and phenols, and majority of the chemical active constituents were found in ethanolic extract. And these are the references. Thank you. The plant material is analyzed for wound healing activity using scratch assay method, and GCMS analysis were also carried out, in which wound healing activity were supported by the presence of active constituents. In ethnobotanically, it is used for various disorders such as beriberi, dysentery, diarrhea, dysmenorrhea, for oral contraceptives also. It is already proven for that diseases, but ethnomedicinally proven wound healing is not scientifically proven. Flavonoid and phenols are mainly active for the wound healing activity. Yes, ma'am. Through phytochemical screening and physiochemical screening, the presence of flavonoid and phenols were confirmed. Thank you.
Good afternoon, everyone. My postal code is 14, and I will presenting my topic on pharmacognostic and phytochemical analysis of tuber Ipomoea mauritiana jack tuber. My plant is Ipomoea mauritiana, otherwise known as giant potato. In Sanskrit, it is known as Vitari. It has various medicinal uses because of presence of various bioactive compound in Ipomoea mauritiana tuber. And its tuber has traditionally used for various purposes such as tonic, rejuvenative, and stimulant. And my aim of my study is to scientifically prove the activities of Ipomoea mauritiana through pharmacognostic and phytochemical screening. The figure 1 and figure 2 are the tuber and flower of Ipomoea mauritiana. Next is the method. After collection, authentication and drying of Ipomoea mauritiana tuber, microscopic and microscopic studies are carried out. Then physiochemical evaluation is carried out, and then phytochemical analysis of the tuber extract is carried. Then next is the result. From macroscopic evaluation, it is externally brownish to dark brownish color, and internally it is creamy white. It has characteristic odor, and it is tuberous, elongated and cylindrical in shape, and 30 to 34 centimeter long, and it has 9 to 10 centimeter wide. It is rough and uneven surface with wrinkles. In figure number 3 and 4 are the TS and powder microscopy of tuber. From this microscopic evaluation, we found that it has various diagnostic features such as cork, bordered bitter vessels, grains, starch grains, and roseate crystals of calcium oxalate crystals. From the physiochemical evaluation, we found that it has low moisture content and total ash value is 4.5. Then from the foreign organic matter, it is below the 1%, and from the formic index, we found that there is a presence of saponin, and from swelling index, we found that the presence of mucilage and polysaccharides, and from the extractive value, the ethanol has more percentage yield, that is 8.5. It represents the presence of polar compound in ethanol. And next is the phytochemical constituent. In the extract of tuber, we found that carbohydrate, flavonoid, phenol, and terpenoid is present in large amount. Next is the discussion and conclusion. Through this study, we found that the tuber is Ipomoea mauritiana, and due to the presence of bioactive compounds such as terpenoid and flavonoid, we found that it has various pharmacological activities. Due to the low value in moisture content and high value in extractive value, we found that it has strong storage potential and it has therapeutic uses also. Through this study, this plant, Ipomoea mauritiana tuber, has further pharmacological investigation can be carried out from these studies. And these are the references. And finally, I'm thanking my management, Nehru College of Pharmacy, and my guide, Dr. G.N. Pramodini, for their support and encouragement. Thank you. So oxalate extraction by using 80% ethanol and with 40 degrees Celsius temperature. Because it contains tuber. It is tuber and carbohydrate is present in it, and from 20% of water is used here. So some amount of constituents are soluble in water, and most of them are soluble in ethanol. So I used both of them for the extraction. Traditionally, it is mainly used for the TB. It is not scientifically proven, and for diabetes mellitus, and to enhance the sperm count and to reduce the pain of body. Sorry, ma'am? Yes, ma'am. Formulation by using suspension.
Good afternoon to my respected judges and my fellow delegates. Myself, G.S. Sudarshan from Sri Ramakrishna Institute of Paramedical Sciences, 3rd year. Today I am going to tell about topic medicinal herbs used in treatment of arthritis. First, let's see the introduction. Arthritis is a chronic autoimmune disease where the immune system mistakenly attacks the joints, causing potential inflammation and leads to joint destruction. Now let's see pathogenesis. First, due to various etiological factors, due to DNA mutation, it causes activation of macrophages, that is anti-inflammatory cytokines like TNF. TNF means tumor necrosis factor, IL-1B, interleukin-1 beta, and IL-NF-KB, interleukin nuclear factor, kappa B light chain enhancer. This how the form is Pannas formation. Pannas formation means the joints, above the joint, the synovial membrane, abnormal tissue will be grown. Due to this, we can see arthritis patients. Next is the crude drugs versus arthritis. The first crude drug we're going to see is turmeric. Turmeric synonymous in Malayalam, we would say mandal, and curcume longe haldi in Hindi. Biological source is the dried rhizomes of curcume longe. Family is Zingiberaceae. The main phytoconstituents are curcuminoids and essential oils, volatile oils, termaron, atlanzone, zingiberone, and methoxycurcumin, dis-methoxycurcumin. Yes, the curcumin is mechanism action. It mainly inhibits the nuclear factor kappa B light chain enhancer because it's a signaling key pathway where it plays a role in inflammation. through this curcumin we can inhibit this. There's a reduction in CRP and ESR. CRP means C-reactive protein and ESR means erythrocyte sedimentation rate. C-reactive protein leads to the rolling inflammation, where erythrocyte sedimentation rate is the diagnostic test for rheumatoid arthritis. Because in patients with arthritis, we can see that red blood cells count in test tube, it will be heavier. Next is the structure. This is structure of curcumin. I'll tell the chemical name. Chemical name is 4-hydroxy 3-methoxyphenyl. 1E6E 1,7 bis 4-hydroxy 3-phenyl 1,7 heptadiene 3,5 dione. 2 ketone groups are there. Because the 2 ketone groups are important for anti-inflammatory and antioxidant activity. Next the drug is ginger. Ginger synonym is inji. In Malayalam we'll say inji, in Tamil inchi. Rhizome, zingiberis and gingerin. The BS is it's obtained from the rhizome of the Zingiber officinale roscoe. The family is Zingiberaceae. The phytochemicals are gingerols, gingiberon and shagols. The 6-gingerol and gingerdione inhibit the 5-lipoxygenase pathway because the pathway leads to the induction. Induction means synthesis of leukotrienes and prostaglandin synthesis. Next, the 8-shagol inhibit the RA means arthritis by targeting a protein called TAC1. TAC1 is the transforming growth factor activated beta kinase 1 because it acts as a central signaling node for TNF, alpha tumor necrosis factor and IL-1 beta, interleukin 1 beta factor, because the two factors are plays a key role in inflammation. Next, 6-shagol inhibit the proliferation of migration, invasion of RA, arthritis, fibrosis, synoviocytes, like matrix metalloproteinase. The matrix metalloproteinase is a group of enzymes that destruct the extracellular joints over the synovial membrane. The structure of gingerols in a bigger shagol. The gingerol is chemical name is 4-hydroxy 3-methoxy 5-hydroxy 3-decanone. The shagol chemical name is 4-hydroxy 3-methoxy dec 3-on decan. Next drug is Indian frankincense. The synonym is in Malayalam we would say Indian kundirikam. Another name is Salai Gugal. The BS, biological source. It is obtained from oleogum resin of Bovelia serrata. The family name is Berseraceae. The phytoconstituents is the bosphoric acids and components are beta bosphoric acid, acetyl beta bosphoric acid, 11-keto beta bosphoric acid. And its chemical classes terpenoids and resin, gum and sugar. Bosphoric acid inhibit the 5-lipoxygenase, the same mechanism for the ginger, and inhibit the production of prostaglandin and leukotrienes. The next is it also inhibits the nuclear factor kappa B light chain enhancer because plays a key role in inflammation. Next, mainly the difference between the ginger and Indian frankincense mechanism is in Indian frankincense it can influence immune response and modulate in the activity of monocytes. The monocytes if it potentiate other immune cells which helps the reduction of inflammation. Next, from research finding we can see these agents provide a valuable foundation in development of safety agents. Thank you. Because there are first 3 classes of drugs, ma'am. So will I choose this? There are many drugs.
Yeah, your presentation is good, Sudarshan. You are from which college?
I'm Sri Ramakrishna Institute of Paramedical Science, College of Pharmacy.
Very good. And you have described about 3 herbs. Okay. So apart from these 3 herbs, can you give spontaneously any 3 herbs which are having same activity against rheumatoid arthritis?
Ginger and Indian.
Gingiber officinalis. Okay. Mike.
The same activity which has the ginger and Indian frankincense, ma'am. Turmeric and Indian frankincense. Because in turmeric, curcumin inhibits the nuclear factor. Also inhibits the nuclear factor kappa signaling pathway. It has same mechanism, ma'am.
Can you name some marketed products out of these herbs? Marketed products which are sold in the market.
No idea, ma'am.
You could have covered that aspect also. Okay,
Extremely
because you are
important,
giving a review article. Okay, if you have given the marketed, then you have covered the nutraceutical products also. Okay,
Extremely
your
important,
presentation
ma'am.
is good.
Thank you, ma'am.
Good afternoon, ma'am, and good afternoon to all my delegates. I'm Pranav. I'm from Sri Ramakrishna College of Pharmacy. I'm studying B.Pharm third year. Now my topic is medicinal herbs used in the treatment of cancer. It's my topic, and I'm gonna say about introduction. Cancer is a multifactorial disease. There are numerous types of cancer are across in a day-to-day daily life. So cancer is a disease which is a significant global health burden. While conventional therapies like chemotherapy, radiotherapy are widely used, they are often associated with some adverse effects which are unavoidable. Recent studies suggest that the certain underexplored or which are going to be explored in future that may have a promise to treat the cancer without with minimal side effects or reduce the effect of chemotherapy and any present going side effects. So pathogenesis. Cancer is occurred from many environmental factors like radiations, chemical agents, and even the genetic factors. They are induced cancer. Carcinogen productions also carcinogen production also and antioxidants. Free radical placements also accumulation in the body can cause also cancer. So this is the pathogenesis of cancer, and inactivation of cancer suppressor gene can helps to reduce or prevention from the cancer disease. So I choose 2 drugs, namely Nigella sativa and Plamago xylankia, which are studied underexplored, which is studied in the in vitro in vivo studies only, not in the clinical trials. They are still exploring the drugs. First one, Nigella sativa, synonym black seed, calzone seed, or black cumin. Biological source, seeds of Nigella sativa, family Ranunculaceae. Phytoconstituents present in the Nigella sativa are thymoquinone, nigellidine, alpha-hedrin, and thymohydroquinone. The chemical structure of thymoquinone is, and the chemical name is 2-isopropyl 5-methyl 1,4 benzoquinone. So mechanism of action of thymoquinone is based on the in vitro in vivo studies, not on the clinical studies. It inhibits nuclear factor kappa B signaling, induces and capsase mediated apoptosis. Capsase is an enzyme which produces apoptosis and suppress angiogenesis and metastasis. Angiogenesis is a formation of blood vessels in the tumor cells and epigenetic modulation of tumor suppressor genes. And Plumbago xylankia, synonym Chitrac and Ceylon leadwort. Biological source, roots of Plumbago xylankia, family Plumbaginaceae. Phytoconstituents present in Plumbago xylankia is Plamangin is an naphthoquinone. Is an naphthoquinone phytoconstituent. It also has a beta-stilbosterol and stigmasterosterol, ferulic acid, gallic acid, ellagic acid, and alkaloids like plumbazilanine. And structure of plamangin is there. Its chemical name is 5-hydroxy-2-methyl-1,4 naphthoquinone. It also inhibits the STAT. STAT is STAT3. STAT3 means signal transduction activation transcription 3. And it generates ROS. ROS means reactive oxygen species, which also it's about the free radicals basics. It also causes DNA damage and leads to cell apoptosis. It also inhibits the angiogenesis. I previously told, no, right? Angiogenesis means the formation of blood vessels in the tumor cells, and also a metastasis. It may act as a chemosensitizers and a radiosensitizers. Research finding. The reviewed herbal agents demonstrated notable anti-cancer properties via selective cytotoxicity, modulation, and transcription factors, epigenetic regulation. Thymoquinine from Nigella sativa showed enhanced efficacy in nanoformulation. While Plamangin from Blumagos xylanthia suppressed the STAT3 in prostate cancer cells, these plants exhibited multi-targeted action, suggesting their potential as agents in the cancer treatment. These agents provides a valuable foundation of the development of safer plant-based treatments. I hope so in future there is a minimal side effects, and it helps to prevent the cancer or treat the cancer with minimum side effect and better bioavailability, and no need to compromise the health. So thank you for giving me the golden opportunity. For my instance, cancer is unavoidable in human in this generation. It's because many other factors like lifestyle modification and use of consumption of over-processed food, ultra-processed food, etc., etc. There's a many reasons. So in future, it could come under the control, not to be in I wanted to be controlled and wanted to be curable. Now I want to be minimize the death. I want to prevent. I want to save the life of the people who have suffered or who will going to be suffer from the cancer. I want to search about underexplored plant because vinclustine, vinplastine, curcumin longa, all those are already discovered and they are going to be formulated. But I want to create a new innovation. I want to provide a setting telescopic pathway. I want to, so future I want to underexplored plants. I want to identify and make it as a formulation and give it to the patient, which I also do not want to compromise the bioavailability of the already existing plants.
Pranav, your presentation is good, and you have given the title as medicinal herbs used in the treatment of cancer, and you have chosen Nigella sativum and Plumbago xylanica. Okay, apart from this, thousands of plants are there which are to be explored. Okay, so in future, you might be having some plant in your mind. Okay, so can you give some other plants by having which are used in treatment of cancer, and any formulations which are available in market, plant-based, which are used in treatment of cancer?
In my mind, I have another plant, and I forgot about that botanical name, and I know other name. Region name. His name is called Anichampu. In Tamil, we have a Tirukural, you may know, right? It's have a one word, one kural, which talk about the anichum. Anichum is a flower, it's have a small fragile petals. It can be shrinked even we smelled. If we smell that petals are very soft and it can be shrink. And this plant may have also an anti-cancer property. It's an antioxidant property. It may hold. Studies are going on about that, and I want to see that plant and I have to smell it. I want to check it. It could shrink or not. That's my one of my bucket list in my bucket list, and I have to. In future, I have an opportunity. I definitely choose that plant and make a study about it. And I could, I hope I could get a better phytoconstituent and make a formulation.
So all the best for your ideology. You have a very good vision. Go ahead with that and develop that plant which you have said, which is given the regional name. Okay, you explore that plant. Okay, all the best.
Thank you for the golden opportunity.
The Nehru College of Pharmacy was established in the year 2003 by the founder Chairman late Shri P.K. Das. Medicinal Garden is a 1-hectare land developed by utilizing the grant obtained from National Medicinal Plant Board. A grant amount of 5,40,000 rupees is obtained for the establishment and maintenance of garden for the time period of 5 years. Medicinal Garden is enriched with 250 plant species which are categorized under 16 subheading. The Herbal Museum of Pharmacy is a dedicated space that celebrates the richness of medicinal plants and their traditional uses. A key feature of the museum is the impressive collection of more than 50 herbarium frames, carefully preserved plant specimens mounted and labeled with botanical names, family, medicinal parts used, and their therapeutic applications. The lab provides a hands-on learning environment for students to identify, analyze, and understand the pharmacognostical and phytochemical properties of herbal materials. Insight 2025 was a dynamic interdepartmental event held on 16 January and 17, 2025 at Nehru College campus. This vibrant two-day celebration marked a unique collaboration among all the streams of the institution, including engineering, MBA, and pharmacy, bringing together knowledge, creativity, and innovation under one roof. The pharmacy exhibition, where students from different departments of pharmacy showcased their innovative projects, models, and research ideas. These exhibits reflected the scientific curiosity and practical insight of budding professionals. The Herbal Expo 2025 was a grand event celebrating the power and potential of medicinal plants, natural formulations, traditional remedies, and demonstration of different extraction methods.
Good afternoon, everyone. Already we are getting late. Now we are move on to the valedictory function. I would like to request Dr. K.G. Prashant sir, Pramodini madam, AO sir, along with the resource persons to be on the dais. Here it's a time to announce the awards for the poster presentation. Best poster award goes to Vaishnavi R, just number CRC 34 from Ahalya School of Pharmacy. I invite Prashant sir along with all dignitaries to hand over the memento. Next we are announcing the first prize for poster presentation, Amrita S. from Ahalya School of Pharmacy. Second prize goes to Surya from Nehru College of Pharmacy. Third prize goes to G.S. Sudarshan from Sri Ramakrishna Institute of Pharmacy. Third prize for Sudarshan from Sri Ramakrishna Institute of Pharmaceutical Sciences. Next, we are distributing the certificates for participants. Any representative from each college come forward and receive the certificates. Ahalya School of Pharmacy. Any representative, please come forward. College of Pharmacy. Sri Ramakrishna Institute of Pharmaceutical Sciences. Anyone from Sri Ramakrishna? Next, Nehru College of Pharmacy. Any M.Pharm students from Nehru College of Pharmacy, please come forward and receive the certificate. we are providing the certificate of appreciation for MPharm 3rd semester 2020 batch. We are appreciating the distinct holders of MPharm 3rd semester from the Department of Pharmacognosy, Srishma K. Babu, Surya S. Nair, Afra P. One by one, please come forward and receive the certificate. Srishma K. Babu, Surya S. Nair, Afra P. Nandita Das, Sri Lakshmi K. Akshaya S. Jasvi Jobi, Fatimatul Nasreen, Mufida, next from the Department of Pharmacy Practice, Josli Jos, Shebiba MP, Shahla, Fatima Abdul Latif, Shebiba, Shahla, Fatima Abdul Latif, Asna, Muhammad Ramshad K. Ranjida Balan, Ramsia Rafi, from the first sem MPharm, from the Department of Pharmacognosy, E. Ashwadi, E. Ashwadi, Fausia K. Akshaya Rajan, E. Ashwadi, Fausia K. Akshaya Rajan. And from the Department of Pharmacy Practice, Nimita K. Rajan. Now we are providing a certificate as a form of gratitude to the organizing committee of this event. I would like to invite Prashant sir along with all dignitaries to hand over the gratitude for the organizing committee. First of all, Dr. K.G. Pramodini ma'am, HOD Department of Pharmacognosy. Mrs. Jesna T.J., Associate Professor, Department of Pharmacognosy. Mrs. Sri Lakshmi K.P., Assistant Professor, Department of Pharmacognosy. Mrs. Drishya E.P., Assistant Professor, Department of Pharmacognosy. Mr. M. Srinivas Nair, AM Nehru College of Pharmacy. Mr. Govind Raj, Assistant Officer Superintendent, Nehru College of Pharmacy. Also, we are providing a certificate for the evaluators of poster presentation. Those who are present here, you can receive the certificate. I will call the names. Those who are present here can receive the certificates. Dr. T. Tamilselvan sir, Head Department of Pharmacy Practice. Dr. D. Sudhakar, Head Department of Pharmaceutical Chemistry and Analysis. Dr. Raju A., Professor, Department of Pharmacology. Dr. Deba T. Vasudevan, Professor, Department of Pharmaceutics. Dr. G.N. Pramodini, Head Department of Pharmacognosy. Mrs. Jasna T.J., Associate Professor, Department of Pharmacognosy. Okay, thank you. So we extend our sincere gratitude to all the organizers from the Department of Pharmaceutics and all the participants, staff from various pharmacy colleges. It's our pleasure to making this event grand success, and it's time too late. Once again, a heartfelt thank you, everyone. Now let's raise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता